I would like to invite the Professor W.B. Gunole sir and Professor D.K. Verma sir for lightening the time and starting the first session of second day of international conference. I also invite Professor Mahesh sir from Rajnandgaon.
बोली भारत माता की जय। सभी खड़े हो जाएं, हाथ जोड़ लें, अपने अपने इष्ट आराधकता को ध्यान करें। ओम अग्नि ज्योति ज्योतिर्ग्नि स्वाहा, सूर्य ज्योति ज्योति सूर्य स्वाहा, अग्निर्वच ज्योतिर्वच स्वाहा, ज्योति सूर्य सूर्य ज्योति स्वाहा। ओम श्री ज्योति पुरुषाय नमः। आवाहयामि स्थापयामि ध्यायामि। ओम गंधाकृतं पुष्पाणि धूपं दीपं नैवेद्यं समर्पयामि। ओम गुरुर्ब्रह्मा गुरुर्विष्णु गुरुदेवो महेश्वरः गुरु साक्षात् परब्रह्म तस्मै श्री गुरुवे नमः। ओम सरस्वती विद्महे पंकजाय धीमहि तन्नो देवी प्रचोदयात्। ओम श्री गुरुवे नमः। ओम श्री सरस्वती नमः। आवाहयामि स्थापयामि ध्यायामि। ओम गंधाकृतं पुष्पाणि धूपं दीपं नैवेद्यं समर्पयामि। फूल पकड़ लीजिए। शुभकामना के सभी फूल पकड़ लीजिए। ओम हम सभी इस कार्यक्रम को हंसते मुस्कुराते खिलखिलाते हुए सम्मानित करेंगे। ओम गुरु ब्रह्म ओम गणनाम त्वा गणपति वं भवामहे, क्रियाणाम त्वा क्रियपति वं भवामहे, निधिनाम त्वा निधिपति वं भवामहे। वशो ममाहं जामि गर्भमातमुजासि गर्भम्। ओम स्वस्ति न इंद्रो वृद्धश्रवः स्वस्ति नः पूषा विश्ववेदाः स्वस्ति नस्ताक्षयो अरसिनेमि स्वस्ति नो बृहस्पतिर्दधातु। ओम पायः पृथिव्यां पायः औषधीषु पायः दिव्यन्तरिक्षे पायोधा पायसति प्रतिशः सन्तु मह्यम्। ओम द्युः शान्ति अन्तरिक्षं शान्ति पृथ्वी शान्ति रापः शान्ति औषधयः शान्ति वनस्पतयः शान्ति विश्वे देवाः शान्ति ब्रह्म शान्ति सर्वं शान्ति शान्तिरेव शान्ति सामा शान्तिरेधि। ओम शान्ति 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 सर्वाणि सुशान्तिर्भवतु। बोली भारत माता की जय। बोली महाविद्यालय सीतापुर की जय। पधारे हमारे सिद्धियों की जय। सरस्वती माता की जय। बोली सूर्यनारायण भगवान की जय। बोली देव देवता की जय। बहुत बहुत धन्यवाद। अपना सूर्य स्थान ग्रहण करने की कृपा करें सभी अतिथिगण।
Thank you, sir. Well, again, welcome back to the second day of this international conference on ICETSTM 2025. I just Dr. Sitapur, organizing secretary of this international conference. Hope you are in great reading and ready for the second day of exciting discussion and insights. Today we have first keynote speaker of today's session is Professor B. Elliot sir, and to introduce him, I would like to invite Professor D.K. Verma sir to. Today we have 4-5 keynote speakers invited talk, and also we have parallel session for the participants, and there is one more poster competition for research scholar, and we are happy to announce there are 2 types of award: best paper award for project, and also we have a best paper award for poster presentation. Our first keynote speaker for today's session is Professor P. Elliot sir from Faculty of Chemistry, National University of Uzbekistan. To introduce him, I would like to invite Professor T.K. Verma sir. Please come on the stage to introduce Professor Elliot.
Thank you very much, sir. Hello, Elliot sir. Am I audible to you? Elliot sir, please confirm. Hello. Hello. Elliot sir, am I audible to you?
Sir.
Hello.
Okay, I will listen you.
Okay, that's good. Thank you, sir. Thank you very much, Professor Elliot, for giving consent to be here between us. And before proceed this program, I would like to introduce our Associate Professor, Professor Elliot Paradee from Uzbekistan. He is Associate Professor at National University, Uzbekistan, Tashkent, and you will be happy to know that he is among top 2% scientists, which is released this year by Stanford University, USA, and Elsevier platform. Professor Elliot has published more than 60 international research papers indexed in Web of Science and Scopus. He has edited more than 15 books. Those are published at highly prestigious platforms such as Royal Society of Chemistry, American Chemical Society, Elsevier, Taylor and Francis, in Springer, and Deepwater. He has H index of 37 and height index of 57. He has completed his PDF from China, and his collaborators are from different parts of globe, which includes China, Saudi Arabia, South Africa, India, Morocco, Turkey, Mexico, Spain, and other developing and developed countries. So, without delay, again, I welcome Professor Elliot Paradee from Uzbekistan on behalf of organizing committee of Government S.P.M. College, Sitapur, Surguja, Chhattisgarh, India. Sir, I welcome you. Mic over to you, sir. Thank you very much.
Okay, thank you for a good presentation, Dr. German. Sir Elliot, I am very happy to join this international conference in India. It is my pleasure to thank you for all persons in this conference. So I am very happy to cooperate with Indian researchers, professors, and so on. So, for example, my best friend and Dr. Verma and other friends in India, they are good personal and good friendly persons. So let me introduce about my presentation. In this conference, I will try to describe little information about corrosion inhibition science and fundamentals, recent trends, and future perspectives. Please next. Okay, it's our country, Uzbekistan. Uzbekistan is located in Central Asia. So we have borders on Central Asian countries, republics, for example, Kazakhstan, Kyrgyzstan, Turkmenistan. Our country is near to India, so we have a good relationship with India. So I would like to continue our friendship in future. Okay, next. This our country is a more historical country. So we and so on. Next. Yes, next, please. This our country is a modern country. If you want to visit our country, you would you would like to see our modern city, Tashkent city. So please next. Just it's a little introduce about our. This is our university, National University of Uzbekistan. Is located in Tashkent city, is capital of Uzbekistan, is our faculty, faculty of chemistry. Our university have a good rankings in times rankings in US higher education rankings. So we have also many several research laboratories. So our government and our university support many researchers in our university. With big funding and research ability. Next. So, what is corrosion? You know, the corrosion is corrosion of metals is a big problem for industries. For example, in chemical industries, gas and oil industries, and so on. Because the main structure element and steel, especially steel, is really active in metals, so it can easily corrode in selenium, medium, aggressive in iron and or neutron mediums. So to protect corrosion is very important. For example, the biggest economic countries like USA, India are involved in China. Around 5 or 3% of GDP is spent to protect metals from corrosion. This move is very big. Okay, next. So I will show the main properties of green corrosion inhibitors. What's the green corrosion inhibitors? Corrosion inhibitors protect metals from corrosion. Now, many scientists attempt to research, especially green materials. In our research, we have tried to synthesize green corrosion inhibitors. These inhibitors also is green and environment also can protect metals from corrosion. There is six main factors. Main factors can support your green corrosion inhibitors. The first one is the greenness. Your materials, if you want to research or synthesize new type of corrosion inhibitors, then following parameters need. The first one is green. Your materials need to be very green materials, biodegradable, non-toxic, not release harmful gases, and very low volatile. The next factor is thermal and chemical stability. It's also very important. The next one, your materials can perform on metal surface. So there is 14 green corrosion inhibitors. First one is ionic liquids, carbon totals, carbohydrates, metal organic frameworks, medicinal, expired medicinal drugs, also green inhibitor, next one, like plant extracts, compounders, and so on. This is for 14 green corrosion inhibitors. It's my suggestion for young researchers. Okay, next. So research methodology. What's research methodology? Why it's important? Because many young researchers don't know how can start, how can finish this research work, which methods may be useful, which parameters need to identify, how about calculation. So there is a pie chart, my suggestion. The first one is making analysis. Then you can identify thermodynamics of activation of corrosion inhibition. The next step is you need to move after gravimetric analysis. You need to go to electrochemical analysis. For example, potential dynamic polarization, electrochemical impedance, electrochemical noise, cyclic voltammetry, S.P.M, electrofrequency modulation, linear polarization resistance. Next one. This is a new type of corrosion inhibitor. Our suggestion, we have used supramolecular compounds. Supramolecular compounds is green materials. So in our research group, we will select new type of corrosion inhibitors on supramolecular compounds. This is main structure of the supramolecular system. Okay, next one. This result is about difference between supramolecular compounds and primary precursors. This one is a cyclodextrin, this infrared structure. Next one, next host, not good molecule orthophenol diamine. This is mechanical mixture, last one supramolecular compound. You can see there is not a big difference in infrared spectra because when supramolecular compounds formed, there is no big changes in the functional groups, just little changes in functional groups. Okay, next one. This one has interesting factors. You can see supramolecular compounds formed. There is not big changes, especially form of functional groups, but very little changes is each atom. Because instead of big extreme, there is 3 types of 5 types of hydrogen, but there is just 2 types of 3 types of hydrogen inside of cyclodextrin is just changing because there is some very strong van der Waals bonds. So you can see these changes on NMR results. Also, it was visible in Raman chapter. Okay, next one. Okay, it is this corrupting mechanism to if you have a question. Okay, metal can corrode. How can corrosion work? Mechanism. So in this presentation, there is 3 types of corrosion mechanism. Corrosion mechanism is very difficult and it depends on many factors. So this is simple examples. For example, you know, as metals easily in the water systems, aqueous water system. Water systems has oxygen. This oxygen is a middle reason to start corrosion of metals. This oxygen is first interacts with hydrogen to form hydroxyl radicals. These hydroxyl radicals interacts with metals. So corrosion product is formed. Okay, next. So it is a pictorial representation of corrosion inhibition absorption. So we will try to describe what's happened of corrosion inhibition and corrosion absorption on the metal surface. So we have advice to this presentation, this mechanism. So there is 4 types of interaction types. The first one, P electrons transfer to D electrons. The next one, electrostatic interaction. The 3 is some P electrons easily transfer D electrons of metals. This last one, free D electrons of metals also can transfer P electrons of aromatic ring. This is a simple representation. Okay, next. So it is not. This is just absorption and translation process. This needs us calculate the thermodynamic parameter. Okay, next. Please next. So. Sorry, back. Okay, this is absorption is not term in our research. So I will advise to use types of absorption, Langmuir, Frunken, Temkin, isotherms. But its best one is Langmuir advised. Okay, next one. So there is results by absorption, isotherms. Main factor is the change of gas energy. The change of gas energy, especially around 20 kilojoule more, it seems absorption is a mixed absorption. It means corrosion inhibitors can absorb on the metal surface by chemical and physical ways. Okay, next. Okay, this cruise is open circuit potential cruise and Tafel cruise. This cruise is important to calculate electrochemical parameters. Okay, next one. This result is from before presentation. You can find these parameters from Tafel properties. So the main thing is a change of corrosion current density. No, then we will it is our corrosion inhibitors to corrosion medium. They can protect metal surface by dramatically reduce corrosion current. Okay, next. This next one is the nitrous and phase angle plots. These plots are very plots by electrochemical impedance results. Okay, next one. So, in the impedance result, transfer resistance is a main factor. Then we will introduce our corrosion inhibitors to corrosion medium. This charge transfer resistance will be decreased. Increase. Because the corrosion inhibitors can protect cathode, cathodic, and anodic electrochemical reactions. As a result, this charge transfer between electrodes is dramatically stopped. So our corrosion inhibitors, it can show how our corrosion inhibitors change and affect corrosion charge transfers between electrons. Okay, next one. This result is cyclic voltammetric grammar of carbon smell and alkaline and saline solution by our inhibitor. So this electrochemical methods is also to show what's happening in reduction potentials and corrosion. Okay, next one. This method, chemical noise results also in interesting. You can also identify how could your corrosion inhibitors protect metal surface by changing electrochemical noise signals. Our corrosion inhibitors, you know, as resistance for electrochemical noise is important. Corrosion inhibitors can importantly increase electrochemical noise resistance. Then you can identify your corrosion inhibitors is very useful or not. Okay, fine. Okay, next one. Electrochemical frequency modulation also interesting to identify how your corrosion inhibitors protect metal surface. Okay, next one. It's the results from S.P.M. Then you can identify how can change cathode in anode excited in corrosion on the metal surface. Unique parts of this method is causal factor. There is two causal factors, factor 2, factor 3. These factors means how can your results is valuable, how can your results more accurate. If your results found causal factors near to 2 is very accurate or near to 3 more accurate. If very low, your results not very high accurate. Okay, next one. So line polarization crew is also important electrochemical analysis. For example, when you add corrosion inhibitors, then resistance is very high. Your corrosion inhibitors can protect metal surface easily. Okay, next one. So this part is surface analysis part. The surface analysis from scanning electron microscope result. And also it explodes. You know, when corrosion product that accumulate on the metal surface, this metal surface is very drastically corroded because there is many drastic cycles. If you add the corrosion inhibitors, there is a thin film formed on the metal surface. Just there is some protective coating formed on the metal surface. Then your material is protected by forming corrosion inhibitor films. The EDKs also show how can change the elemental spectra after protection. Okay, so next. This Raman shift on the metal surface before corrosion. Red line is before corrosion. No. Red is corrosion. This green is after protection. How can change on a functional glucose after protection? So it was for some signals like ferrium, iron to nitrogen signals formed after protection. These results confirm protection performance of corrosion inhibitors. Okay, next. So atom forces also interesting to identify surface morphology with metal surface before protection and after protection. Average error factor is also to show what happened before and after corrosion. Now, in protection systems, average roughness is very low, so corrosion can protect metal surface. Okay, next. This analysis is theoretical analysis. In theoretical analysis, we have theoretical analysis have two parts. The first one is the density functional theory part. Next one is molecular dynamic simulation part. In DFT analysis, we have identified some key parameters of molecules. For example, LUMO and HUMO regions, difference between LUMO and HUMO, and chemical softness, chemical hardness, chemical potential, dipole moment, and so on. So all parameters show how can your corrosion inhibitors interact with metal surfaces. Especially chemical absorption depends on whole region. While physical absorption also depends on LUMO region. Okay, next one. This is the results also HUMO results from this other methods from Gaussian. Gaussian is also interesting. In this result, we will identify what the changes, what the difference between neutral inhibitors molecules and protonated inhibitors molecules. Because when you if your corrosion inhibitors to corrosion medium, if this medium more acidic, then your corrosion inhibitors protein acid, then form other structure. So what's happened? And it was found this protein corrosion inhibitors can get effect in the neutron inhibitor molecules. Okay, next. So this pictorial representation of corrosion inhibitors, how interact with metal surface. The simulation system is contained water molecules, acidic molecules, and metal surface. It was found it was found this difference, this chemical bonds around 3 angstroms. 3 angstrom it may be 3 angstrom it may be this chemical bonding system. Okay, next one. This also the same results. Okay, next one. This also same results by Monte Carlo methods. Okay, one minute. Okay, sorry, one minute. Okay. Okay, this one is if the results also show this size of chemical bonds, and the length of the chemical bonds between chemical compounds and metal surface. So this shows some molecules can interact with metal surface by chemical absorption. Someone is physical absorption. Okay, sorry, sir. Okay. Next one, please. So this is a corrosion inhibition mechanism. It's over suggestion. This over inhibitor. So there is properties of chemical absorption and chemical absorption mechanism. There is 4 types of chemical bonding. The first one is a corrosion inhibition corrosion mechanism. This one is formed very stronger H plus in chlorine minus ion. Also hydrogen gases. These gases also to form. In inhibition medium, the first step inhibitor can protect it to form protected compound. This protected compound maybe interacts with metal surface, also interacts with chlorine ions. As lastly, this formed compounds can protect, can absorb on the metal surface. As a result, metal surface is protected, it's non-corrosion products. Okay. So, as a conclusion of my presentation, I will show there is a three types of factors very important to identify how about identify the corrosion inhibitors may be efficient or not efficient. The first one, you need to know environmental stimulation, immersion time, corrosion inhibition, concentration, solution, temperature, metal tape, maybe iron or copper and so on. Media tip is also important. Maybe your media is acidic medium, saline medium, and iron and so on. Next one, your corrosion inhibitors be more hydrophilic and hydrophobic properties. Also can absorb on the metal surface. Absorption properties also very important. The last properties, corrosion inhibitors maybe need to be more specific hydrophobicity compound. Also, these compounds have many heterocyclic rings, aromatic rings, and alkyl changes, and many functional profile in alkaline changes. This properties is main reason for to create more inhalatory green corrosion inhibitors. Okay, next. Okay, next, please. Okay, next, please. Okay, this is okay. The first one next. Okay, please. Okay, it's our research team, Department of Physical Chemistry. Okay, next one. Okay, so it's the main region of things for our purpose in this conference. I am very like to join your conference. Next time I will go to India to attend your conference by online, by not online but offline. Okay, thank you.
Professor Elliot, am I audible to you?
Okay.
Sir, am I audible to you? Hello?
Yes, I am.
Sir, thank you very much, sir. The way you
Okay, thank you.
Sir, the way you have delivered this complicated topic in a very simplest manner, and we have your audience, those that belongs to UG, PG, and research scholar, in which includes professors also. This is definitely a global problem. Corrosion is a very global problem, and the way you have systemized and systematically present your lecture, and definitely I think it will be a much way to our scholars, our professors. Sir, on the behalf of our country, India, and on the behalf of organizing committee, on the behalf of organizing institute, government, S.P.M college, and most important, from the behalf of institutional head, principal, professor, sir, I convey my gratitude, my thanks to you for giving your valuable time to us. Sir, thank you very much. You can leave, sir. Hello? Okay, sir.
Yes, yeah, hi.
Thank you, sir. Thank you for the confirmation. Our next keynote speaker is Professor Srikant S. Manda, sir, Associate Vice President, Dr. HEQ Lab, Newcomb Informatics and Visiting Scientist, Cancer Data Science, Children Medical Research Institute, Sydney, Australia. We welcome you, sir, for the keynote lecture on the topic of the NGS revolution, multi-omics approaches to understanding health and diseases. Thank you, sir. Thank you, sir, for joining. Over to you, sir.
Yeah, so am I audible? I'm audible, right?
Yes, sir. Yes, you are audible, sir.
Thanks a lot for inviting me. I think the lecture has been, the conference has been going very well. So just to start, I would like to thank Dr. Rohit Barga for inviting me for this, and the organizers, especially Dr. Topo sir, for organizing this nice event. So what I'll try to do, so my forte has been on analysis and other on next-gen sequencing data. So I can share my screen, right?
Yes, sir. Yes, sir. Please share, sir. Now it is visible, sir.
Is it in full screen?
Yes, sir. Yes, sir. Now it is in full screen.
Okay, yes.
Thank you.
Yeah, so what I'll try to go in the next 15-20 minutes is the NGS revolution and how it has helped us understand the different health and diseases. So, to start the very basic, because there are a lot of students out here, so I would like to start in saying the DNA, so that we know that the DNA is the genetic material that consists of 4 nucleotides, adenine, cytosine, thymine, and guanine. So there are various sources of this DNA. You can get from where you can actually get the DNA, which is shown in the figure here, like it can be bones, hair, tooth, blood, saliva, and these are very commonly used resources. Coming to the topic, that is next-gen sequencing. So what is next-gen sequencing? So it has all started with the Sanger sequencing, which is very first in early 1970s, which was started by Frederick Sanger, wherein we sequence the nucleotides of interest. So there is one base, so every base, the complementary base adds in, and then you are able to detect that base. So that was called the Sanger sequencing, but there were limitations on that, where you could only go up to 500,000 base pair fragments, and it was also very costly. So that's when the revolution of the next generation sequencing has come. So that was the first generation. So anything that came out after the generation is the next generation sequencing. So the next generation is where you have parallel sequencing, so you can sequence multiple nucleotides, so the DNA fragment which I showed. So you isolate the DNA and then you sequence them, many molecules at a time, which has given the power of sequencing multiple genomes. So you can now do like the entire plant genome or animal genome or microbial genome, you can sequence entirely. So there are machines like the Iron Torrent, Illumina and others. I'll go briefly on that in my later slides. And then recently there has been another set of sequences which are called the third generation sequences. So these are all related to long read sequencing, so ultra long and long read. So you can sequence, so earlier when it was 500 bases, now you can actually go up to MBs, like 1 to 10 MB of the entire genome. So if you have like small virus or bacteria that you want to sequence and know the composition of the nucleotides, you can actually have only one read that can cover the entire sequence. So that is the revolution that has happened from first to second to third generation. Coming to the history, if you see, like I was saying, so 77 was when the Sanger sequencing method was developed, but later as the new method started coming, the first human draft genome which came in 2001, it took almost like from 1990 to 2001 for them to actually come up with one human genome sequence. But now with the new sequences like the Illumina, which was launched in 2006 and others recently, within one day you can actually sequence the entire human genome. So 3 billion bases can be sequenced in like one day. So that is the revolution that has come. Coming to the different platforms, so there are different platforms, and the concept behind majority of them is, so you take the genome of your interest, so you isolate the DNA, you fragment them with some enzymes, and then you put some adapter sequences at the end, and then you amplify them. So how you amplify is the way like different machines are there. So there are the major players in this are companies called Illumina and Pacific Biosciences and Thermo Fisher. So Thermo Fisher works in by detecting the proton, the H ion that is released whenever there is a nucleotide incorporated. The ligation assay is another way where you can ligate base and then detect that, and then you can also do something called as bridge amplification. That is also one of the methods. So overall, the idea is that you have a base of your interest, you add one base from outside. If it is a complementary, you attach some kind of fluorescent probe to it, and you can detect that probe. So that is how most of these techniques work. So this one is a bit complicated slide, but I'll try to make it easy. So what all things you can actually do if you do sequencing? So routinely we are doing very lab experiments and other things. So if you have a DNA or RNA from the sample of your interest, whether it is plant, animal, or soil, or any kind of things, so you can actually take and study the genome. So the genome can be studied. If you want to study the entire genome, then it is whole genome sequencing. If you are studying only some region, only the coding regions, then it is whole exome sequencing. If you want to study only specific genes, then you do a targeted sequencing. If you are studying the entire micro, like you want to know what are the bacteria, fungi, virus in a given sample, so you can do a metagenome sequencing. And then if you are interested in understanding the methylation, like what is happening at the epigenome level, so you can do an epigenomics. On the RNA level, again, if you are interested in knowing the gene expression patterns in any given sample, so you can do whole transcriptome. And within that also, if you are interested in, let's say, small RNAs, so microRNAs, so you can study that using sequencing techniques. So you can do a snRNA or a long non-coding RNA, so which are RNAs which are greater than 200 base pairs. So those all techniques, those all kind of analysis is what you can do using next-gen sequencing. So as I was mentioning, so what are the applications? So in terms of applications, so you have the complete genome sequence, you have the metagenome, you have transcriptome, you have microRNAs, then you can also do ChIP-seq and other different techniques. So these are some of the machines that you can utilize. So at Nucleome Informatics here, which is based out in Hyderabad, so we have most of these machines and we do routinely sequencing. So if you have small applications, like if you have some plant or animal genome that you want to sequence, you want to study some microbes, so then you can use these machines like MySeq, NextSeq and others, or from other companies like Thermo Fisher, there is IMG, Studio, X5 and GenXS. If you have bigger genomes, like human genomes or mouse and those kind of things, if you are studying, so you can use machines like NovaSeq 6000, NovaSeq X series, so these can do up to like, so if you see the data output, so it can generate up to 16 TB of data. And you can get the data within like 48 hours of the analysis. So the entire analysis you can finish in like 2 days. So that's the how powerful these long sequencing techniques are. And then the recent modern ones, which are called long read sequencers. So we have two major players here, Oxford Nanopore and Pacific Biosciences Review. So if you see in this table, so the read length goes anywhere from 10 KB to 1 MB. And accuracy is also very high, and you can finish these runs like in 24 hours. And you can do a lot of different things, like if you're interested in understanding copy number changes, any structural variations in the data, if you are looking at repeats in the genome, so you can use, mostly we are using it for clinical genomics. In terms of human genomics, if you are studying, so that is one of the areas which we work a lot. So we see that this you can use to study the chromatin accessibility, the structural variations, repetitive regions, and it's also used to understand the non-coding regions of the genome. So if there are a lot of non-coding regions now, there are a lot of studies that are coming up which says that non-coding region of the genome has a very high role. So that also you can study using this sequencing. So how a typical next generation sequencing analysis works out is, so you start up with sequencing and then you get the data. So you have a reference genome, which is basically a genome of your interest. So let's say I sequence a plant genome, which is not available. I have a reference for that, and then I will actually align to that reference. So there are tools. So these are all computationally intensive tools. So you need to have good computational power for doing this analysis. And then we identify the variants that are there. And then based on the variants, you can actually define whether they are coming. So we do it routinely for cancer genomes and others. So you can tell it whether it's coming from a cancer or it's a normal sample. So those kind of things you can do. And then we also prepare reports based on that. Coming to multi-omics, so when the term, because I put that multi-omics, so usually what we are seeing is nowadays people don't stick to just one omic data set, like they will not do just genomics or transcriptomics. So you have multiple omics like genomics, epigenomics, transcriptome, proteome, metabolome. So based on, so what you can do is for a given sample, you can do more than one omic data sets, and these are on the top like things that you can identify using each of the home. And then what you do is you merge all the data together to find something which is co-regulatory. So and this is very much useful in studying things like cancer, because there only one thing is not changing. So you see that there are methylation changes, there are transcriptome changes, there are genomic changes. So that is very much needed for understanding. And in terms of applications, so you can see that there are a lot of applications that you can use for with the multi-omics data sets. So you can do tumor normal comparisons, you can do enrichment analysis. So you can actually look at expression. So if you have some sample of your interest and you want to see under stress and without stress, so what is happening? So you can actually look at that with the multi-omic data. And then you can also look at if you have more than what are the different subtypes, what are the different kinases that are there, what are the acetylation that is happening. So you can do a lot of different analysis if you have more than one different omic data sets. So coming to our area of interest, which is precision medicine, and this, so the term precision medicine comes in from, so earlier most of the treatments that happened for any patients was the traditional medicine approach, which says, so let's say you you go to the doctor and say that I have a headache. So what they'll do is they'll give you a tablet, and which is like the general tablet for anybody. But what recently has been seen is this doesn't work. Like maybe some people actually that same medicine works, for some it doesn't work. So why is it that? That is where the precision medicine comes. So we see that each individual has their own genomic architecture. So based on your DNA, based on your architecture, actually we can design specific therapies and medicines so that it works very well for you. So it works on 4 different aspects, which we call a P4 medicine: personalized, predictive, preventive, and participatory. So it helps you to identify things that are specific to a given. So we call it the precision medicine approach. And what are the key drivers for that? So the key drivers are the new machines which I was just discussing. So the high throughput data that can come out. So you can now, and then there is so much of data available in terms of, so we all use now like Facebook, Twitter and all social media data. So your information is there everywhere. Then there is the environmental data, so based on which place of India you are residing. That also has a lot of effect on your genome, epigenome. And then you have the digital data. So all this data along with like the new revolution, that is the AI, so artificial intelligence. So these have actually triggered, made the key drivers which actually help in the precision medicine approach. So you have to put all this data together to understand like what is specific for a given person or an individual or a given group. So it could be like, so that we have seen like there are people from northeast of India, so they respond differently to a given medicine versus people from south of India. So that is also because of the way what they eat. So one side of the India they eat, let's say, rice, other side they eat mostly like roti and chapati. So you see that their architecture, the genomic happening actually works out like that. So that we need to know specifically like how it works. And to give an example, in cancer especially, like why it is important, so that is one area which we work extensively. So we see that like there are two, for example, there are 2 lung cancer patients, same stage, same histology. But you see that one person actually works very nicely with the drug, he will have remission, the cancer is gone, but the other person has basically not remitted. So why is that? That is because of the genomic architecture. So unless you understand the genome, it is very hard for you to know that what happens and how actually to treat that particular person. Same thing like I was saying, like the medicine. So if you give a normal drug to somebody and then they respond, but some of the other people may not, and that is because of the genomic architecture. And that particular makeup, which is actually very much important in any of the cancers, and for studying these cancers, we specifically need to understand, like if you want to understand the inheritance pattern, if you want to know, like if there is a family, in all of them have diabetes in the family, so you would like to know, is it actually genetic in them, so that in the next generation, like you can monitor and then ensure that that person doesn't get diabetes. And this one example. So how it works, the approach, very briefly I will quickly go through. So you have a group of patients or a group of people. So you collect the sample from them, and then you do these different omics which I was describing, like you can do metabolomics, transcriptomics, other things. And using that you identify markers or biomarkers which are specific for each patient. So now you know that there is a molecule which is high in this patient group, blue. There is one which is high in this brown, one is green. So what you do is you give only that particular treatment against that for this group, for the Y, for the other group, and for the other different. So what you see is now you have precision medicine approach where precisely it is working only for a set of people, and that would actually help much better to start. And this is one approach which is being used now globally. So we have been working extensively, like with Children's Medical Hospital in Sydney and other places where we are trying to find cure for most of these childhood cancers and others. So in the company where I'm currently located in Hyderabad, so we have all the different machines. So if based on your interest, you can actually do the sequencing. So we do sequencing as a service, and then we can actually provide you the analysis and everything. So whether it is for whole genome, RNA, or whichever sequencing method which I just described. We also recently started something called wellness. So just by your DNA profile, we can tell you like what is the good diet for you, like nutrigenomics. So you can tell what to eat, what is good to eat, and what is not good to eat. How does your vitamin D levels are good, vitamins. So a lot of things you can do just from knowing your DNA or genetic makeup. And in terms of the other research that we are doing, so we offer a lot of different services, and I am actually heading this group which works on analyzing the data from different services. So yeah, so you can always contact me if you have any interest in knowing more about this. So coming quickly to, so how am I with the time? I have time, right?
5 minutes. 5 minutes.
Okay. So quickly I'll go. So I'll just show one study which recently we have. So I know I am not much aware on the audience on the cancer side, but I'll quickly try to show. So there are 3 challenges in cancer genomics which you might be hearing a lot. So one of them is metastasis. It's like the cancer is at one place, like it metastasizes and goes to different place. So it's very hard to detect from where it has come. The other is resistant. It's like a patient has been given treatment and then you see that after some time he stops responding. And the third one is relapse. So the doctor said that it's all good, you're cancer free, go home. He came back after 2 months again, he got back the cancer. So these 3 challenges are very key to any of the cancer that occur, and we are trying to address this through multiple approaches. So what happens is actually these mutations, so as the cell divides, they keep on accumulating mutations. So they keep on getting new mutations, and once they have enough mutations that basically can change the cell, that is when the cell becomes a cancerous cell. So it is a very random process, so you cannot say that like I didn't have any habit, I never had anything, but still I got cancer. So that is because the cells keep on dividing and then the division results in that. So you need to understand these differences in the cells, which is called the heterogeneity. So we see that one place, let's say there is a lung cancer, and that cells in that lung cancer, there are different tissues. So that they actually go into a different place, and then if you don't understand the entire composition of that, it's very hard to treat those patients. So on that line, so we did a recent study on ovarian cancer. I will just quickly show you something. So this was recently published in Nature. This is with Westmead Institute of Medical Research in Sydney. So what we tried to do is, this was for the high-grade serous ovarian cancer. So that is very common epithelial cancer, even in all women, even in India as well. So what happens, the patient is, by the time they go there, it's already spread to other parts. So it happens in the uterus, but the ovarian cancer then spreads to other parts, and then it is very hard to treat. So there are ways to treat. So they will remove that, or they use some inhibitors in others like PRAGA, and there are a lot of like drugs that are available. But what happens is after some time this recur, and some people actually respond, some don't respond. So what we try to do is to find out, is there a set of genes or set that we can use and differentiate these patients. So for that we just did a study in which we took 11 different patients and did all the different omics that I was describing. So we have done RNA sequencing, we have done next-gen sequencing, we have done proteomics, we have done genomics on them. And we tried to put all of them together to see, is there anything that is stably there in all these samples, and can we use that to come up with some kind of drug. So the interesting thing, one of them I'll show. So this plot here, this shows each of the color is like samples taken from one patient. So you can see the patients, whatever sample you take, they all come together. So that is one thing, like within the patient, you can see like it is very tightly grouped, but each patient is different. So you need to find out like things that are there which is common, then only the treatment will work for the entire group. So using some pipelines that we developed, so I'll not go through that pipeline now because of the time. But what we have come up with, we came up with a 50 gene signature, and this signature can be used to differentiate the patients, and that is specifically related to the immune pathway, so immune system. So what happens is this, if you actually find these genes and their signature in any of the patients, so this triggers a pathway called the STING pathway. And this pathway basically can be used to trigger the immune system. So you can actually target those genes, and then it will elicit the immune response in the patient, and then the patient who was not responding earlier, they will start responding. So that is one interesting study. So and just to show one of the example I will put here. So each patient, if you see, so there are different things. So there is a DNA damage in the patient because of the cancer, and then we see that the expression of mRNA, these are multi-omic data sets, and then we also see the expression of this PD-L1, which is required for the tumor recognition. So we can actually individually find out like patients that are working or not working, and then so right now the study is further going on. So we are trying to come up with the drugs that can work on these patients. It's a bigger cohort they are collecting now. So I think I'll just quickly round up. So this is a very famous slide which says that the cancer is like a hydra. So it's a 9-headed hydra. So hydra is a mythical creature. So it's like if you cut one head, two more comes up. So cancer is something like that. So unless you have something which is the multi-omics, which can cut all the different heads, then only you can actually come with a personalized cure or a personalized care for this patient. So I hope I am able to convince them the need and what is precision medicine and why it is needed. I think, so these are my acknowledgements, and my contact is here. So if you want to contact me, please let me know. And I'm thankful to the organizers and the committee members for giving me this opportunity. Any questions, please let me know.
Any doubts, any questions? Students? Sir, I think we have no queries. Thank you so much, sir.
Yes.
Thank you, Professor Manda. On behalf of our institution, Govt S.P.M College Sitapur, and organizing committee of this international conference, I would like to extend heartfelt gratitude to you, sir. Your lucid explanation on this next generation sequencing lecture a field that is rapidly transforming genomics and biomedical research was exceptional. And once again, I thank you, Professor Manda, for taking time out for your busy share. Thank you, sir.
Thank you. Thank you very much for inviting me. Yeah, thanks.
For the next technical session, we have Professor Prashant Tiwari, Department of Pharmacology, College of Pharmaceutical Science, Dayanand Sagar University, Bangalore. He will deliver his lecture on innovations in neuroendocrinology towards personalized medicine and endocrine disorders. session for this was Professor Prashant Tiwari, but he's not available due to some technical issues. Now we have our keynote speaker, Dr. Rashmi Verma from Dr. C.V. Raman University, Bilaspur. I request Madam to deliver her lecture on public awareness on environment through environmental protection. Environmental education. Welcome, ma'am.
Very good morning, one and all. Respected Dr. Nurul sir, Rohit Barja sir, dignitaries from other college, dear students and scholars. I am Dr. Rashmi Verma, Associate Professor and Head Department of Chemistry, Dr. C.V. Raman University. So today, the topic of my presentation is public awareness on environment through environmental education. Because I am working in environmental pollution, my research area is that. So that this topic is very relevant to my research. And first of all, I want to take this chance to talk to you. Make this two-way communication, not one-way communication. Because each and everyone used to do presentation that is one way, so you can't relate with such presentation. So I want to make this presentation two-way. Okay, so as you are already seeing, आप देख रहे हैं कि environment day, every day is our day when your home is melting. So much message in this clip. आप समझ पा रहे होंगे कि इसमें कितना बड़ा मैसेज है कि अगर आपका घर टूट रहा हो और आप अर्थ डे एक दिन 22 अप्रैल को मना रहे हैं, तो दैट इज नॉट गुड. ईच एंड एवरी डे शुड बी अर्थ डे. हर दिन आपका अर्थ डे होना चाहिए. तो ये क्यों है, ये तो आप अगर सोचें तो जस्ट हम बोल रहे हैं हम पॉल्यूशन में हम पार्टिसिपेट कर रहे हैं, और जब हम ये कहते हैं कि हम पॉल्यूशन कर रहे हैं तो हम अपने साथ 4 लोगों को जोड़ते हैं, एंड वी आर टॉकिंग अबाउट इन जनरल नॉट पर्टिकुलर। सो आज से आप कहिए कि मैं हूँ पॉल्यूशन के लिए जिम्मेदार। आई एम द वन हु इज रिस्पॉन्सिबल फॉर पॉल्यूशन, हु इज रिस्पॉन्सिबल फॉर ग्लोबल वार्मिंग, हु इज रिस्पॉन्सिबल फॉर ओजोन डिप्लेशन। आपको पता है जो पॉल्यूशन है वो ग्लोबल वार्मिंग, जो आपका समय ठंडी देर से आ रही है, रेन बहुत दिन तक चल रहा है, गर्मी बहुत ज्यादा पड़ रही है, तो ये सब क्या है? डिस्टर्बेंस है। और सब कुछ आप अभी तक सुनते आए हैं कि हमारी वजह से, लेकिन सब कुछ है वी नहीं, I am the only one who is responsible for such type of pollution। जितने तरह की प्राकृतिक आपदाएं आ रही हैं, विपदाएं आ रही हैं, जो भी हो रहा है, सबके लिए हम रिस्पॉन्सिबल हैं। So moving forward, why environment? So I am focusing कि why environmental education is implemented in higher education. What is the need? और जब हम पढ़ते हैं तो हम क्या करते हैं? Environment तो है, कौन क्लास अटेंड करेगा? कितने बच्चे हैं जो इन्वायरमेंट की क्लास को सीरियसली लेते हैं? कोई भी नहीं। क्यों नहीं लेते हैं? क्योंकि हम इसको लिख लेते हैं, ऐसे सोचते हैं, लेकिन उसके पीछे उद्देश्य क्या है? व्हाट इज द एम बिहाइंड इम्प्लीमेंटिंग एनवायरमेंटल एजुकेशन इन हायर एजुकेशन? क्या जरूरत पड़ी? तो क्या है? क्लाइमेट चेंज इज द एनवायरमेंटल चैलेंज ऑफ दिस जनरेशन एंड कमिंग जनरेशन, एंड इट इज इम्परेटिव दैट एक्ट बिफोर इट्स टू लेट। हमको एक्ट करना पड़ेगा। जितने भी हम का रिसर्च कर रहे हैं वो सब सोसाइटी बेनिफिट तभी है, इट इज सोसाइटी बेनिफिशियल ओनली इफ इट इज रिलेटेड टू एनवायरमेंट, बिकॉज जितनी भी प्रॉब्लम, जितनी भी समस्याएं आने वाली हैं, सब एनवायरमेंट की वजह से ही आने वाली हैं। द एनवायरमेंटल क्राइसिस इज ग्लोबल प्रॉब्लम एंड ओनली ग्लोबल एक्शन विल रिजॉल्व इट। आपने ये 2012 में देखा कि कैसे कौन सी जगह है ये, कोई बताएंगे बच्चे? मैंने पहले कहा दिस शुड बी 2-way communication, not 1-way communication. So you all need to participate in my lecture. So क्या है ये, कोई बताएगा बच्चा ये क्या है? कभी आपने रील स्क्रॉल करते करते कुछ देखा है? केदारनाथ फ्लड, तो आपने सर्च किया है क्यों आया था? तो ये हो गई प्रॉब्लम जो सिर्फ और सिर्फ एंथ्रोपोजेनिक एक्टिविटी के कारण आई। व्हाट इज एंथ्रोपोजेनिक एक्टिविटी? मैन मेड रिसोर्सेज। अभी ये दिल्ली का गार्बेज का जो माउंटेन है, माउंटेन ऑफ गार्बेज, पूरा कचरा का गार्बेज, और एक बार ये गार्बेज साथ से चल रही हुई ट्रैफिक पे गिर गया था और बहुत बड़ा एक्सीडेंट हुआ था दिल्ली में। अभी कुछ इसमें रिजॉल्व कर रहे हैं उसको, लेकिन फिर भी अभी रिजॉल्व नहीं हुआ है, और ये एक प्रॉब्लम है। ये तो अभी आप न्यूज में देख ही रहे होंगे, अगर आप न्यूज देखते हैं कि अभी दिल्ली में AQI 300, 500, तो AQI क्या है? एयर क्वालिटी इंडेक्स, प्रेजेंस ऑफ गैसियस पॉल्यूटेंट एंड सस्पेंडेड पार्टिकुलेट मैटर इन कंपोजिशन ऑफ एयर, दैट इज air quality index. This is pollution in river Yamuna in Delhi. Now some action were taken to resolve it, but more work is needed. This is Chennai water crisis. Chennai is going to be dry city. Chennai dry city almost हो चुका है। और आपने जब क्रिकेट अगर आप फॉलो करते होंगे तो जोहान्सबर्ग में जब हमारी इंडिया टीम कुछ साल पहले खेल रही थी, तो वहां पर भी उनके नहाने और के लिए पानी खत्म हो गया था, अगर आप फॉलो करते हैं। ये बेंगलुरु सिटी है, सिटी ऑफ लेक्स, और यहाँ पर सिटी ऑफ बर्निंग लेक्स, जो वहाँ पे फॉग है और जो झाग है उसमें आग भी पकड़ लेती है, इसलिए उसको बर्निंग लेक्स बोलते हैं। और ये बहुत बड़ा प्रॉब्लम है लेक्स का, और लेक्स अब ड्राई भी हो रहे हैं वहाँ पे। सो इतनी सारी प्रॉब्लम मैंने आपको इसलिए बताई कि you must relate with that. Why environmental education is important? Why? Let's make our world a greener हरा बनाएंगे, दैट इज इम्पॉर्टेंट। सो आप इन्वायरमेंट एजुकेशन क्यों एजुकेटर्स को ये लगा कि इन्वायरमेंट एजुकेशन इम्पॉर्टेंट है, और एनईपी भी कहता है अभी वैल्यू एडेड कोर्स में इम्प्लीमेंटेड है इन्वायरमेंटल एजुकेशन एंड सस्टेनेबल लिविंग, तो वहां पर इसको इम्प्लीमेंट किया गया। और सिर्फ दिस इज नॉट अ पर्टिकुलर एम टू just put the syllabus and curriculum to just you have to pass this particular course or particular paper. It is one action because of you have to know about how pollution occurs and what are the remediation and what are the process to be done to resolve it. Understanding the need. Environmental education is not collection of environment information related to environment. मतलब ऐसा नहीं है कि इकोलॉजी क्या है, आपका इकोसिस्टम क्या है, ग्रास इज ईटेड बाय ग्रासहॉपर इज ईटेड बाय फ्रॉग इज ईटेड बाय स्नेक इज ईटेड बाय हॉक एंड हॉक ये लास्ट कंज्यूमर प्रोड्यूसर, ये सब नहीं है इसका मतलब है कि आप प्रकृति को जानें, आप एनवायरनमेंट से कोरिलेट करें और आप समझें कि कैसे इस पे आगे काम करना है। और रिस्पॉन्सिबिलिटी किसको लेनी है, आप जोर से बोलिए, किसको लेनी है रिस्पॉन्सिबिलिटी? मुझे, नॉट हम। जब आप हम बोलेंगे तो आपकी रिस्पॉन्सिबिलिटी बढ़ जाएगी, आप सोचेंगे अरे मैं भी और मेरा बगल वाला भी, मेरी सहेली भी, मेरा दोस्त भी, मेरा भाई भी, सब। लेकिन जब आप रिस्पॉन्सिबिलिटी को बांटेंगे नहीं और अपने ऊपर लेंगे, तो आप अगर एक चिप्स और एक कुरकुरे का पैकेट भी खा रहे हैं तो आप उसको फेंकने को क्राइम समझेंगे कि इसको मैं अगर रोड पे चलते हुए फेंक रहा हूँ तो दैट इज क्राइम। ओके, इट प्रोवाइड्स फैक्ट अबाउट एनवायरमेंटल इशू एंड टू मेक द जनरल पब्लिक अवेयर अबाउट एनवायरमेंटल इशू। तो आप पढ़ेंगे, आप अपने घर में बताएंगे और जनरल पब्लिक तक ये मैसेज जाएगा कि एनवायरमेंटल इश्यूज क्या होते हैं और कैसे होते हैं। The devices through which environmental problem are highlighted and brochures, bulletin and video are made through other techniques. इसमें ये है। अब प्रॉब्लम कौन कौन से हैं तो ये बताने की जरूरत नहीं है। एयर पॉल्यूशन, क्लाइमेट चेंज, ओजोन डिप्लेशन, आपका स्ट्रेटोस्फीयर है, वहाँ पे ओजोन लेयर है, वो क्या करता है, वो अल्ट्रावायलेट रेज को धरती पे आने से रोकता है। फिर अगर इसमें डिप्लेशन होगा, अगर उसमें क्षरण होगा तो क्या होगा, अल्ट्रावायलेट रेज एज कहीं पे वहाँ पे होल है तो वहाँ से अल्ट्रावायलेट रेज नीचे आएंगे और सबसे पहला इफेक्ट क्या होगा, राइज इन सी लेवल, और सी लेवल राइज होगा, पोलर आइस कैप्स मेल्ट होंगे और सेवरल इफेक्ट ऑन मैन एंड इट्स वेजिटेशन इज आल्सो। आउटडोर पॉल्यूटेंट्स, इंडोर पॉल्यूटेंट्स, नॉइज इसमें ऐड हो गया है। वाटर pollution any foreign material, that presence of any foreign material in composition of water which depletes the quality of water for man and its vegetation is known as water pollutant। तो water pollutant में क्या है, आपका DO सबसे ज्यादा, जब डिसॉल्व ऑक्सीजन जब कम है तो क्या होगा, बायोलॉजिकल ऑक्सीजन डिमांड बढ़ जाएगा, आपके पॉल्यूटेंट बढ़ जाएंगे। आप पानी को बहुत दिन तक रख दीजिए तो उसमें से स्मेल आती है क्योंकि उसमें ऑर्गेनिज्म बढ़ जाते हैं और ऑक्सीजन कम हो जाता है। तो ये एक तरह का पॉल्यूटेंट है। टॉक्सिक केमिकल्स जो हैवी मेटल्स हैं, ऑर्गेनिक पॉल्यूटेंट है, दैट आर टॉक्सिक एलिमेंट जो आपको डिजीज भी देते हैं और क्रॉनिक डिजीज भी देते हैं। पेस्टिसाइड इनडायरेक्ट चैनल ऑफ वाटर पॉल्यूशन है। पेस्टिसाइड जब आप क्रॉप लैंड में पेस्टिसाइड जाके मिक्स होता है, उसके बाद जब रेन वाटर में मिक्स होता है फिर वो वाटर रिसोर्सेज को जाके पॉल्यूट करता है। वेस्ट प्रोडक्शन, सबके घर से वो वेस्ट निकलता है, सॉलिड वेस्ट, हजारडस वेस्ट मतलब जैसे हॉस्पिटल का वेस्ट है जिसको सॉइल वेस्ट भी कहते हैं। और ये सारी प्रॉब्लम, सॉइल वाटर लॉगिंग, कोस्टल पॉल्यूशन, सो मेनी प्रॉब्लम। Importance of environmental education, approaches by which people develop awareness. This is not for just implementing particular course. This is for awareness on people and generation. Initiative कब लिए गए environmental education को implement करने के लिए? The first Earth Day on 22 April 1970 अर्थ डे हुआ और उस अर्थ डे में इस पर डिस्कस हुआ कि कैसे वाटर पॉल्यूशन को रोका जाए। और 1992 में रायो डी जेनेरो में भी आज समिट हुआ, जहां से मेन फोकस आया कि इन्वायरमेंटल एजुकेशन को हायर एजुकेशन में पुट किया जाए। द यूनाइटेड नेशन एजुकेशन साइंटिफिक एंड कल्चरल एजुकेशन ऑर्गेनाइजेशन यूनेस्को, यूनाइटेड नेशन यूएनईपी, यूनाइटेड नेशन एनवायरमेंटल प्रोग्राम created 3 major declarations that have guided course of environmental education. What is UNEP? UNEP क्या है? The United Nations Environment Program is an agency of United Nations and coordinates its environmental activities, assisting developing country in implementing environmentally sound policies and practices. उसी का एक है कि सिर्फ 5 जून को आप environment day मनाते हैं, और वो environment day यूएनईपी और यूनाइटेड नेशंस के हर बार उसका थीम आता है एक पर्टिकुलर, उसके बेसिस पे वर्ल्ड वाइड सेलिब्रेट होता है। मिशन टू प्रोवाइड लीडरशिप एंड एनकरेज पार्टनरशिप इन केयरिंग द एनवायरनमेंट बाय इंस्पायरिंग, इन्फॉर्मिंग एंड एनेबलिंग नेशन, दैट इज मोस्ट इंपॉर्टेंट मिशन। गोल्स टू इम्प्रूव द क्वालिटी ऑफ एनवायरनमेंट, आप पढ़ेंगे तभी तो एनवायरनमेंट की क्वालिटी को इम्प्रूव करने के लिए आपको इंस्पिरेशन मिलेगा, और वो दैट इंस्पिरेशन इज मोस्ट इम्पॉर्टेंट फॉर दिस एनवायरमेंटल, फॉर ओवरकमिंग द एनवायरमेंटल पॉल्यूशन, टू क्रिएट अवेयरनेस ऑन एनवायरमेंटल पॉल्यूशन, एनवायरमेंटल प्रॉब्लम एंड कंजर्वेशन, टू क्रिएट एन एटमॉस्फेयर टू डेवलप कैपेबिलिटी, टू इवैल्यूएट डेवलपमेंटल प्रोग्राम फॉर एनवायरमेंटल प्रॉब्लम्स। तो ये अलग अलग प्रोग्राम और ये सब समझने के लिए एनवायरमेंटल एजुकेशन का गोल है। ऑब्जेक्टिव ऑफ एनवायरमेंटल एजुकेशन, अवेयरनेस फॉर एनवायरमेंट एंड इट्स रिलेटेड प्रॉब्लम। अवेयरनेस, जो वेरियस डेज मनाए जाते हैं, ओजोन डे, अर्थ डे, वाटर डे, बायोडाइवर्सिटी डे, वाइल्ड एनिमल डे और इन्वायरमेंट डे, ये सब अवेयरनेस के लिए किए जाते हैं। फिर दूसरे नॉलेज, ताकि आपके पास नॉलेज रहे कि क्या करना चाहिए, जैसे इन्वायरमेंटल पॉल्यूशन कब हो, क्या उसके रिमेडियल प्रोसेस है, व्हाट आर द रिमेडियल, सो दैट यू हैव सम नॉलेज अबाउट इन्वायरमेंट। क्योंकि आप जब छोटे में वो पढ़ेंगे तो वो उतना आपके साथ नहीं रहेगा। जब आप हायर एजुकेशन में आते हैं तो आप एक एज को पा जाते हैं जहाँ से आप हर कुछ समझ आपकी प्रॉपर बनती है, अंडरस्टैंडिंग बन जाती है। एटीट्यूड टू एक्वायर मोटिवेशन, आप मोटिवेट होंगे तो एटीट्यूड अपने आप आ जाएगा। स्किल, रीमेडियल प्रोसेस के लिए स्किल देना पड़ेगा कि कैसे रीमेडियल है, क्योंकि सब बता देंगे कि ये पानी पॉल्यूटेड है, ये सॉइल पॉल्यूटेड है, एयर पॉल्यूटेड है, दिल्ली में इतना सब बता रहे, लेकिन अभी वहाँ पे कृत्रिम वर्षा, आर्टिफिशियल रेनिंग कराई जानी थी लेकिन नहीं हो पाया। बहुत सारे प्रोसेस हुए, IIT कानपुर ने भी किया लेकिन नहीं हो पाया। तो स्किल होना चाहिए कि कैसे ओवरकम करेंगे। और पार्टिसिपेशन, आपको इतने जो मैंने डे बोले उसमें जब आप पार्टिसिपेशन करते हैं तो आप एक इनिशिएटिव ले सकते हैं कि अपने आसपास आप जाइए। जैसे हमारे बच्चे डिपार्टमेंट ऑफ केमिस्ट्री के बच्चे सीवी रमन यूनिवर्सिटी में जाते हैं, आसपास की जितनी भी एरिया है सबको डस्टबिन लेके आते हैं और बताते हैं कि जितना भी प्लास्टिक है आप इसको इसमें डालिए, और फिर उसके बाद वो जो व्हीकल है, जो कलेक्शन व्हीकल है जो म्यूनसिपैलिटी से आता है, उसमें आप दीजिए। कोई भी कचरा आप सामने मत करिए, और आप अपने घर में करके देखिए, जब भी कोई प्लास्टिक का कचरा है तुरंत उठाइए। कॉलेज में भी आप क्या कीजिए, कोई गुटखा का पाउच फेंक के जाए, बिल्कुल उठाइए, आपको उसमें कोई भी वो नहीं होनी चाहिए। आप जैसे वो करते हुए दिख जाएंगे, आपके जो सफाई कर्मचारी है वो आपसे पहले उसको उठाएंगे। तो ये है पार्टिसिपेशन। The evolution of environmental education in global perspective. So the concept of environmental education was first initiated by International Union for Conservation of Nature (IUCN) in 1970. A meeting in Nevada, USA. STEM introduced. उस समय STEM introduce हुआ और 1992 में भी वो implement हुआ। So ये क्या है आपका evolution process कैसे होगा, curriculum में क्या होगा, and what are the steps to be taken. This is why UNESCO UNEP 1976 में ये implement हुआ। फिर Belgrade Charter 1975 was developed in as United Nations Education, Scientific and Cultural Organization Conference in Yugoslavia, and it is widely accepted goal statement for environmental education. वहाँ से एक statement आई, the main aim of environmental education is to develop a world population that is awareness and concerned about environment. मतलब जो पढ़ रहे हैं वही तो awareness फैलाएंगे और उन्हीं के अंदर तो concern होगा कि कैसे environmental pollution को रोकना है। अब आते हैं मेन हमारा environmental education in Indian perspective. So in India, environmental education has been given importance because lots of effort been already initiated to achieve sustainable development for last several decades. So international agreements such as Agenda 21 called a reorientation and of all education towards sustainability, UNCED 1992 chapter, that is I am already talking about Rio de Janeiro conference, the R Summit. Providing environmental education at different levels of education, primary education में क्या होता है? Awareness of environment and knowledge only. Secondary education, relevance for real life situation of environment, understanding बने. Higher secondary education, conservation of natural resources of environment. College and university, sustainable development. This is most important point. Sustainable development by solving problem of environment. So attitude and evaluation. ये most important aim है higher education का. अब UGC में क्या initiative लिया? तो the decision UGC mandates environmental study in UG level. The decision to create 6-module course on environmental study was taken by Supreme Court in 1991 at that time. After a public interest filled by Advocate M.C. Mehta, according to the court order, UGC had prescribed a course on environment and mandatory subject in college. This is UGC initiative. Environmental education in curriculum process, learning about environment, learning for environment, learning through environment. This is most important curriculum process. तो उसमें ये तीनों चीज आनी चाहिए curriculum में, जो भी curriculum design करते हैं इसके लिए, तो ये सारी चीजें. Key component of curriculum planning process. So active orientation towards development of skill rather than imparting information only. College level में सिर्फ information आपको नहीं देनी है, skill के लिए भी आपको उस syllabus में जगह है. और यही NEP 2020 में भी जब environment डिजाइन हो रहा था environment का syllabus, तो वहाँ पर भी ये सबसे important point था कि active orientation towards the development of skill rather than imparting information only. Knowledge should shape into both soft and hard skill. Soft skill भी होना चाहिए. अभी AI आ गया, एक slide है मेरा यहीं पे, AI and hard skill, और hard skill भी होना चाहिए. Development of positive attitude, value and appreciation towards the nature. Method of environmental education, field-based education, interactive classroom session, service learning education, learner-centered education, group projects, problem-solving activities. Problem-solving activities मतलब एक केस स्टडी करना है और फिर उसकी प्रॉब्लम को कैसे सॉल्व करना है. रोल ऑफ टीचर, अब करिकुलम तो हो गया, अब टीचर का क्या रोल है? The role of teacher in environmental education. So, education acts as powerful tool in socio-economic transformation of a society, community, nation. So, teacher is main person for that. So, teacher should be skilled. Some attitude towards environment must be in teacher. So, behavior towards children or learner or student to promote desire of environmental awareness and resolving also. The need to have educator who will able to teach environmental education, both formal and non-formal education program, and further implement this program called for the training of teacher in teacher education. So teacher must be trained. ऐसे नहीं कि उसको environment है तुम पढ़ा सकते हो, ऐसा नहीं. जिसको भी environment subject दिया जाए उसको प्रॉपर ट्रेनिंग दी जाए कि हाउ उसके अंदर ही शुड हैव स्किल, एटीट्यूड and are also aware about all the problems related to environment and all the information related to environment. So teaching methods, discussion method होने चाहिए, demonstrate, कुछ प्रोजेक्ट दे दिए, कुछ मॉडल बनाने के लिए दे दिए, डेमोन्स्ट्रेट हुआ, प्रोजेक्ट हुआ, किसी फैक्ट्री में भेज दिया, जाओ देख क्या वहाँ का एग्जॉस्ट कैसा है, वहाँ एग्जॉस्ट में एब्जॉर्प्शन मेथड, कंडेंसेशन मेथड, कंबशन मेथड है कि नहीं है, वहाँ पर ग्रेविटी सेटलिंग, फैब्रिक फिल्टर, इलेक्ट्रोस्टैटिक प्रेसिपिटेटर है कि नहीं है, तो उसको रिजॉल्व करते हैं। डिस्कशन है, वो बच्चों के साथ बैठे और डिस्कस करें कि क्या है प्रॉब्लम, और एक एक चीज को जब आप जीडी करते हैं तो ज़्यादा इफेक्टिव होती है। मीनिंग ऑफ डिस्कशन, तो एक्टिविटी पर्सन जो है टॉपिक को इनिशिएट करे और बच्चे फिर उस पर अपने थॉट्स रखें। इंपॉर्टेंट ऑफ डिस्कशन मेथड, टीचर इनिशिएटेड डिस्कशन, स्टूडेंट इनिशिएटेड डिस्कशन, पैनल डिस्कशन और सिम्पोजियम, बोथ, ये चारों। और जीडी टॉपिक्स जो होंगे, लीकेज इन वाटर पाइप्स, दैट इज मोस्ट इंपॉर्टेंट प्रॉब्लम, सीवेज पाइप इशू ऑफ लाइसेंसेस टू कंस्ट्रक्ट टॉल बिल्डिंग, वाटर पॉल्यूशन इन सी, डिस्पोजल ऑफ सी में कैसा वाटर पॉल्यूशन, तो ऑयल स्पिल आपने सुना होगा, मुंबई पोर्ट में ऑयल स्पिल हुआ था और वो बहुत बड़ा प्रॉब्लम था जो रिसेंट में हमने फेस किया था। तो आपको ये सब होना चाहिए, और आप जितना न्यूज़ सुनेंगे, न्यूज़पेपर पढ़ेंगे, उतनी आपको नॉलेज भी बढ़ेगी और आप इस सारी चीज़ों को रिलेट करेंगे, तो आपका करिकुलम आपको कभी भी टफ नहीं लगेगा। डेमोन्स्ट्रेशन मेथड इज ऑल्सो मोस्ट इंपॉर्टेंट मेथड, तो आप करके दिखाना है कि कैसे रिजॉल्व हो रहा है। प्रोजेक्ट मेथड, प्रोजेक्ट बना के बच्चे, ये तो होता ही है, प्रोजेक्ट बना के लाए प्रॉब्लम पे और उसको तो जब बच्चा उसको बोलेगा तो उसको अपनी ज़िंदगी भर वो उसकी मेमोरी में रहेगा और उसको याद रहेगा कि मैंने अपने बीएससी फाइनल ईयर में ये प्रोजेक्ट बनाया था, तो उसकी एक एक चीज़ उसने डेमोन्स्ट्रेट की थी और एक्सप्लेन की थी, तो वो उस नॉलेज को एक्वायर करके जब प्रेजेंटेशन देगा तो वो सारी चीज़ें समझेगा। द प्रोजेक्ट मेथड इज प्रॉब्लमैटिक एट carried to completion in its natural setting, that is project. Purpose, activity, utility, freedom, feasibility. ICT tool in environmental education, so all the ICT tools, communication, information and communication technology, digital tools यूज करके अगर environment वीडियो दिखाया जाए, वीडियो लेक्चर, वर्चुअल लैब्स दिखाए जाए, जो आईआईटी का, अभी हमारा CD रमन में आईआईटी का चैप्टर भी है वर्चुअल लैब का, तो हम भी बच्चों को तो वर्चुअल लैब से भी दिखाते हैं सारा कुछ, तो वो वर्चुअल लैब बच्चों को दिखाना चाहिए, वहाँ से भी उनको रीमेडिएशन प्रोसेस, जो वर्क ऑलरेडी डन शुड बी डेमोन्स्ट्रेटेड बाय वर्चुअल लैब। यूज ऑफ इंफॉर्मेशन टेक्नोलॉजी, यूज ऑफ जियोग्राफिकल इंफॉर्मेशन सिस्टम, डेटाबेस, इन्वायरमेंटल इंफॉर्मेशन सिस्टम, रिमोट सेंसिंग, वर्ल्ड वाइड वेब, ऑनलाइन लर्निंग सेंटर, टीचर फ्रेंडली फीचर, स्टूडेंट फ्रेंडली फीचर। यूज ऑफ मीडिया एंड टेक्नोलॉजी इन एनवायरमेंटल एजुकेशन, तो ये मोस्ट इंपॉर्टेंट है। मैं बार बार बोल रही हूँ कि मीडिया, न्यूज, तो जितनी चीजें मैंने बोली मैक्सिमम न्यूज में आप देख सकते हैं, आपके इंटरेस्ट की है, तो हम न्यूज नहीं सुनते हैं कि सिर्फ उसमें बहस हो रही है, सिर्फ उसमें पॉलिटिकल इशू हो रहे हैं, लेकिन आपको न्यूज सुनना चाहिए कि क्या क्या प्रॉब्लम्स हो रहे हैं। So how can we control environmental pollution? So green investment है, solar इलेक्ट्रिसिटी है और इको फ्रेंडली टेक्नोलॉजी है। एफॉरेस्टेशन में, डिफॉरेस्टेशन मतलब फॉरेस्ट का नष्ट होना और एफॉरेस्टेशन मतलब फॉरेस्ट को प्लांटेशन करना, फॉरेस्ट को रिस्टोर करना। एजुकेशन एंड ट्रेनिंग। एजेंसीज फॉर इन्वायरमेंटल प्रोटेक्शन, तो ये दीज आर सम नेशनल एजेंसीज, ERT, CSE, EFI इन्वायरमेंटल फाउंडेशन ऑफ इंडिया, HWO हिमालयन वेलफेयर ऑर्गेनाइजेशन, विद्वान इकोलॉजी एंड नेचुरल हिस्ट्री ऑफ फाउंडेशन, VENF, ये सब इन्वायरमेंट के लिए काम करते हैं। इंटरनेशनल एजेंसीज, ESGP अर्थ सिस्टम गवर्नेंस प्रोजेक्ट, ग्लोबल ग्रीन ग्रोथ इंस्टीट्यूट GGGI, इंटर गवर्नमेंटल पैनल ऑन क्लाइमेट चेंजेस से ये सब काम करते हैं। EEA यूरोपियन इन्वायरमेंट एजेंसी, यूएनईपी, ये सबसे ज़्यादा इन्वायरमेंट के लिए काम करते हैं, यूनाइटेड नेशंस इन्वायरमेंटल प्रोग्राम। लिस्ट ऑफ इन्वायरमेंटल एजेंसीज, Environmental Protection Agency, Field and Fish and Wildlife Service, Botanical Survey of India, Indira Gandhi National Forest Academy, National Park Agency, Greenpeace, Worldwide Fund for Nature, the Bombay Natural History Society, Kalp Vriksha Society, Kerala Sanskrit Sahitya Parishad. Environmental movement कब कब हुआ? बिश्नोई मूवमेंट 1700, चिपको मूवमेंट 1973, सेफ साइलेंट वैली मूवमेंट 1978, जंगल बचाओ आंदोलन 1982, एपिको मूवमेंट 1983, नर्मदा बचाओ आंदोलन 1985, टेवरी डैम जो है डैम कॉन्फ्लिक्ट 1990। इंपॉर्टेंट एक्ट्स, अब आपको एक्ट्स भी पता होना चाहिए। तो इंडियन फॉरेस्ट एक्ट जो फॉरेस्ट कंजर्वेशन के लिए है, एनवायरमेंटल प्रोटेक्शन एक्ट, Indian Forest Conservation Act to provide act may be called Forest Conservation Act, मतलब फॉरेस्ट को कैसे सेव करना और कैसे रिफॉरेस्टेशन करना है। Wildlife Conservation Act, Biodiversity Act। अब अभी nowadays after 2014 स्वच्छ भारत अभियान, Clean India and Green India Mission, National Green Tribunal, Center of Environmental Studies, इकोवास, लेजिस्लेटिव रिफॉर्म, नमामि गंगे परियोजना, स्वच्छ भारत अभियान। स्वच्छ भारत अभियान के लिए मैं आपको इंफॉर्मेशन देना चाहूंगी कि ई स्वच्छ पोर्टल है, जहां जाके आपको वीडियोस देखने हैं जो इंडिया में इन्वायरमेंटल प्रोटेक्शन के लिए जो स्वच्छ सिटी बनाई जा रही है उसके लिए, और फिर आपको उसके 25 क्वेश्चन के आंसर देने हैं। फिर आपको स्वच्छ भारत अभियान की तरफ जब आप 100% आंसर देंगे उसके बाद आपको एक सर्टिफिकेट मिलेगा स्वच्छ भारत अभियान से, जो आपके लिए बहुत सारे बच्चों के लिए इम्पोर्टेंट हो सकता है। तो आप आज ही उसको गूगल कीजिए, ई स्वच्छ पोर्टल। इम्पोर्टेंस ऑफ एनवायरमेंटल एजुकेशन, तो ये मैं बार बार आपको बता चुकी हूँ। क्रिटिकल टूल इन 21st सेंचुरी, हेल्प एड्रेस टू नेचर डेफिनेट डिसऑर्डर। How can we control environmental pollution? Conclusion. Environmental education, इतना मैंने बताया, उसका कंक्लूजन यही है कि आपको अवेयर बस नहीं, इंफॉर्मेशन बस गैदर, नॉट ओनली गैदरिंग इंफॉर्मेशन, यू हैव टू वर्क ऑन रीमेकिंग, दैट इज मोस्ट इम्पॉर्टेंट इंफॉर्मेशन by UNEP or other agency towards environmental education, which is already implemented from many years in higher education, that is motto. But this students take this not seriously, because वो सोचते हैं कि यार ये परीक्षा देके हम तो खुद से ही लिख लेंगे environment क्या है और एयर पॉल्यूशन क्या है, तो कुछ लिख देते हैं, people wash their dirty buffalo in hot water and it is called water pollution, तो ऐसे आंसर लिख देते हैं और इतना सीरियसली लेते हैं। तो आपको सीरियस लेना है एनवायरनमेंट, इसलिए दैट्स व्हाई दिस इज इम्प्लीमेंटेड इन योर करिकुलम। दीज आर सम रिकमेंडेशन कि ये सब डेज मनाने चाहिए सारे एजुकेशनल इंस्टीट्यूशन में, तभी ये अवेयरनेस होगा, और अवेयरनेस हर प्लेटफॉर्म पे होना चाहिए। दिस इज माई सर्टिफिकेट आई एम आस्किंग, जब आप खुद करेंगे तभी आप सबको बोलेंगे कि ये करने के लिए, तो ये मेरा सर्टिफिकेट है। जब आप 25 के 25 आंसर देंगे तब आपको ये सर्टिफिकेट होगा। तो आप ये सर्टिफिकेट अगर लेना चाहें तो ई स्वच्छ पोर्टल में जाकर रजिस्ट्रेशन कीजिए, और 25 एक जैसे गिफ्ट सिटी गुजरात का आपको 30 मिनट का वीडियो दिखाएगा, और उस 30 मिनट के वीडियो से आप उसको फिर जाके क्वेश्चन आंसर देंगे, और जब 25 आउट ऑफ 25 आप मार्क करेंगे मार्क्स, तब आपको ये सर्टिफिकेट आएगा स्वच्छ भारत अभियान की तरफ। लेना चाहेंगे आप लोग? करेंगे गूगल? थैंक यू। अब एक ये वीडियो है जो मैं प्ले करना चाहती हूँ। सर वो फोन दीजिए तो पास में। एक मिनट में वीडियो प्ले करूँ। आएगा आवाज? अच्छा यहाँ है? जी। हाँ जी। आपके वहाँ पे
टिक 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 कसम ये खाए रे टिक 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 प्लास्टिक टिक ना पाए रे टिक 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 कसम ये खाए रे टिक 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 प्लास्टिक टिक ना पाए रे अगर सुन लो जो संभलना हम तो बम ये फट जाएगा ये बहती सी नदियां का ये पानी रुक जाएगा
अगर सुन लो जो संभले न हम तो बम ये फट जाएगा कि बहती सी नदियां का ये पानी रुक जाएगा
कि दर्द रहे धरती का ये दम भी घुट जाएगा
कि कुदरत की नस नस में जहर ये बस जाएगा प्लास्टिक के हमसे मोहताज है कैसा ये समाज है कल ना हो हाथों में आज है
मौका ये अपना बनाए टिक 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 कसम ये खाए रे टिक 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 टिक
प्लास्टिक टिक ना पाएंगे
टिक 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 कसम ये खाए रे टिक 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 प्लास्टिक टिक ना पाए रे सिचुएशन ड्रास्टिक है जहाँ देखो प्लास्टिक है सिचुएशन ड्रास्टिक है जहाँ देखो थैली बोतल स्ट्रॉ इत्यादि जर्रे जर्रे में फैलाने ना घट पाए ना मिट पाए रह रह के टॉक्सिक बन जाए इससे छुटकारा तो नहीं पर है एक सरल उपाय रे सिंगल यूज प्लास्टिक को हम कह दे बाय बाय रे टिक 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 टिक
कसम ये खाए रे
टिक 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 प्लास्टिक टिक ना पाए
there any questions, any doubts?
और मैं
कोई
क्लास
प्रश्न पूछना चाहता है?
और मैं क्लास में बोलूँ तो अगर आपको डाउट नहीं है तो फिर मेरे को है।
Very nice presentation on the environmental education. One thing I want to clear that you have shown the video also on plastics. So what is the remedy? What is the solution for plastic?
Sir, plastic
It creates many problems.
single-use plastic,
And,
microplastic.
So what to do now to avoid plastic? Is there any remedy or any solution?
So, sir, we have to work on biopolymers. Instead of
What type of biopolymers? We want clear.
Some research have to be done regarding making
But
biopolymers.
now at present the plastics are there everywhere. So how to minimize these things?
Sir, सबसे पहले तो first of all we don't want to use, we don't need to use plastic, single plastic. We have to carry some other things, steel bottle or
Okay,
other things.
you are
That
correct.
is not-
Single plastic is there. Everywhere the bottles are same. But how to
plain
minimize it?
plain
How to-
water should be replaced by this should be replaced by plain water. And this is our initiative, but researchers should be more serious about doing research on biopolymer instead of the polymer.
I don't know that these plastics are not degradable. We want things which are to be biodegradable.
Biodegradable.
So
That is why
is there any compound which is biodegradable?
Sir, I'm already told about biopolymer. So there are several things making polymer by starch or from the bio.
Do you know the polymers
Yes, sir.
which can be used for biodegradable plastic?
There is, you can take some bio product or bio-based से आप polymer बना सकते हैं। और फिर उसमें सर आप some compositing also there filler nanofiller use कर सकते हैं। nanofiller use करके biopolymer की property को use कर सकते हैं। इसमें भी भरपूर है।
And what about the environmental pollutions?
Environmental pollution is everywhere and it should be researchers should work on remediation instead of on negative.
For example, air pollutions.
Air pollution is a separate process and we have already.
Now what is the alternative? Pollutions are what are the alternative? What to do for it?
First, sir, pollution should be cured before it evolves or increase something.
No, I don't know that automobiles pollutions are air
Yes,
pollutions
sir.
are there.
Some
Industries are there.
processes should
Household
be implemented
materials are there.
exhaust
So many
system.
things are there. What are the alternative for that?
Sir, exhaust system should be updated and put some adsorbent and some condensation process and some process should be which is used to remove of air pollutants such as substituted particulate matter is different and gases pollutant is different, so that exhaust system should be advanced and should be capable of removing that gases pollutant such as oxide of carbon, oxide of nitrogen, oxide of sulfur, hydrocarbon and sulfated particles.
What are the factors affected for the climate change? What are the, tell me the example, the factors which are affected for the climate change?
Sir, global warming, ozone
Which factors are responsible?
anthropogenic activities.
So this is one. Many factors are there.
Many factors are there, man-made sources, natural sources, other sources are there.
Now, I want to clear that what are the what are the things, name of the things by which that the climate changes are there. But there is a global warming.
Global warming, the property of Earth which
Yes, it is changing. Yes, global warming is there, but what are the factors again? Which part is responsible?
Greenhouse gases. Which are
Not only one.
Sir, what is global warming? That is the property of Earth by which it retains heat. And that is presence of certain gases.
Why the warming is there? That's what I'm asking.
Sir, because increasing greenhouse gases and most contributed greenhouse gases is
This is okay here, only one. Okay. carbon dioxide.
Sir, so many processes are there. So many processes.
For example?
Reforestation is main process. कि आप पेड़ लगाइए और फिर converted in carbon oxide converted in other things and exhaust से remove करें, remove from exhaust.
Okay.
Any other
अभी तक पर्यावरण प्रदूषण के केवल बाहरी पर्यावरण प्रदूषण के संबंध में चर्चा की गई, जिसका समाधान हम देखते रहे, समाधान में हम असफल रहे हैं। अभी तक जितने भी समस्याएं गिनी जाएगी केवल बाह्य पर्यावरण के संबंध में गिनी जाएगी। हमारा प्रश्न है कि बाह्य पर्यावरण का सोल्यूशन खोजते खोजते हम थक जा रहे हैं, फिर इसका समाधान अभी तक नहीं है। स्वतः की ओर से यही है कि अगर आंतरिक पर्यावरण को प्रदूषण को देखें तो इसका समाधान संतुलन हो जाएगा और इसका हल उसी में। इस संबंध में आप क्या कहना चाहेंगे?
जैसे हाउस होल्ड वेस्ट है, किचन वेस्ट है, अगर हम उसको डिस्पोज ना करके घर में उसकी कंपोजिटिंग या सोसाइटी में वाइट बना के कंपोजिटिंग करेंगे तो शायद डिकम्पोजिंग वेस्ट को हम रोक पाएंगे, जिससे वो पॉल्यूशन जो है रिजेक्ट फॉर्म होकर जो वाटर सोर्स का यूज होता है वो एक ये प्रोसेस।
इसमें हम यही पाए कि जितने भी समस्याएं हैं वह पर्यावरण से संबंधित हैं। आंतरिक पर्यावरण के शुद्धिकरण के लिए शायद यह हमारा सेमिनार प्रयास करे तो संतुलन हो सके। हम रिस्पॉन्सिबल कब होंगे?
जब हम अपनी रिस्पॉन्सिबिलिटी लेंगे, अभी तक तो हम ले ही नहीं। कोई अगर ये नहीं कहते जैसे मुंबई IIT की कर रही है सोलर प्लांट का सोलर बस, लेकिन वो बस एक ही व्यक्ति ना जोड़िए अवेयरनेस के, हम तो आप किसी का नाम नहीं सुनते।
शायद महाविद्यालय परिवार जितने हमारे
आपने सुना
पधारे हैं सभी
IIT के सर जो लिखे हैं सोलर बस के, वो अवेयरनेस कर रहे हैं और किसी का नाम बताइए जो अवेयरनेस कर रहा है, मैं भी नहीं करती।
हम सबको करना चाहिए, नहीं करना चाहिए।
Thank you so much, Dr. Rana. Your lecture was not just informative, it was a powerful call to action. And definitely education plays a dominant role in transformation of socio-economic changes. I once again, on behalf of our organizing committee, I thank Dr. Rashmi Verma from Dr. CB University, Kota, Bilaspur, on her valuable, informative and fruitful lecture. Thank you, ma'am. Now we have our invited talk for Professor Prashant Tiwari. He is available now online. Professor Tiwari, can you hear me? Am I audible, sir? Hello, Dr. Prashant Tiwari, am I audible? Dr. Tiwari, can you hear me?
Yeah, madam, I can hear. Very good morning.
Good morning, sir, and warm welcome to our international conference. For next technical session, we are having Professor Prashant Tiwari, Department of Pharmacology, College of Pharmaceutical Science, Dayanand Sagar University, Bangalore. I request Professor Tiwari to deliver his lecture on innovations in neuroendocrinology towards personalized medicine and neuroendocrine disorder. So without wasting a time, I request Dr. Tiwari to start his lecture. Thank you, sir.
Yeah, thank you so much, madam, for your deliberation, and wishing you the best wishes and congratulations for this great event. I would like to share my presentation. Can I share with you now, sir? Okay, let me share. From my side, I am facing some issue. I am sharing my PPT to Rohit sir. Please, can you share, sir? So there are some technical errors from my side. I have shared my PPT to Dr. Rohit sir. Could you please share, sir, if possible? Thank you so much. Please, could you make it in a presentation mode? Yeah. Thank you so much. I'm very sorry for the inconvenience. So it is really a great matter and pleasure to have this international conference organized by the government SHYAMA PRASAD, MUKHERJEE College, Sitapur. I'm going to deliver a session on the innovation in the neurological disorders towards the personalized medicine and endocrine disorders. As we knew that recently there is a requirement of the personalized medicine for the multiple disease and disorders. In addition to that, neurodegenerative disorders require a lot of attention. Neurodegenerative disorders are characterized by multiple diseases, for example, Parkinson, Alzheimer's disease, Huntington's, etc. If you could see the physiology and the background behind these endocrine disorders, so here there are many people who are suffering with multiple neurological disorders followed by metabolic disorders. So Alzheimer's is a disease. It can be characterized by the loss of memory, which is also known as dementia. For such cases, we require a personalized drug and medications to handle the specific disease based on the biomarkers. Those biomarkers are known as neurotransmitter agents, for example, dopamine, acetylcholine, GABA, epinephrine, norepinephrine, etc. And those biomarkers can be easily determined and estimated by blood sample, by collecting the plasma samples, where we could be able to find out a level of certain neurotransmitters, and we could see how a technology intervention can be involved to take care of the neuroendocrine disorders. Next, please. In addition to that, you could see if there are a certain exciting therapy and personalized medication. If we could involve the artificial intelligence or machine learning to address those personalized errors that may be due to the genetic modulation or genetic alteration or lifestyle modification or food habit. So in such cases, we require a personal care to address certain neurological disorders. If there are errors over the medication effect or maybe the course of administration, it could be recorded through the system and it can be noted immediately to address certain medical emergency. If you could see, there is an generational gap and it could be transmitted from one person to another person, and it may lead to changes in the mutation of tissue. Nowadays, there are multiple changes on our lifestyle modification due to lifestyle modification, food habit, etc. It is altering the genes and it is increasing the certain neurotransmitters or endocrinological disturbances, which leads severe disorders. Suppose it may increases the prolactin level more than the 10 AUG in the plasma, which is known as the hyperprolactinemia. Hyperprolactinemia is a very severe disorder now. It could be characterized by gynecomastia, galacturia, loss of derivate, lot of alterations are happening. It can also affect the thyroid level, folic acid hormones, and this PSH is responsible for the cognitive behavior. So in such cases, to address certain personal based error, it is required to address those neurological disorders. Next, please. So there are a system where we can identify those data on a real-time basis. So those real-time data can be addressed through the computational screening or maybe the molecular docking. So this molecular docking is very simple. We should not make complex. There is a set of tools, there is a set of data, and we have to identify a specific protein which is a response, which can bind with a specific drug or maybe the tissue or maybe the cell, where their association can be increases with the protein, higher the binding affinity towards the specific site, higher will be the responses. So this is the advanced tool where we can address those ambiguity and unclear data to be coded and addressed for the certain neuroendocrinological disorders. That is nothing, that is the neurodegenerative based symptoms. Here a set of tools or data can be screened and fitted over the system, and we have to address a protein or a specific gene. For example, there is a DYRK1A genes. Due to the overexpression of this DYRK1 gene, there is a modulation over the neurological disorders like Alzheimer's disease. So those are the neglected adverse effects of certain antipsychotic drug. For example, haloperidol 0.1 mg per kg administration or 0.5 mg per kg administration. So this haloperidol works based on the dose dependency. It may lead to dependency of the particular cell or the gene, which leads and occurs to manipulate or to alters the gene's expression, which can be addressed through the advanced technology by the intervention of the talking of in silico study. So for that, there is a Surguja software which could give us a precise data or targeted disease or gene where we could involve this personalized medication, or we can manage the error over the medication. Next, please. So in addition to that, if we wanted to advance in our system with the help of AI or with the help of talking study, we have to identify certain factors which are responsible for development of certain disease. So we have to see the drug interaction. So their interaction can be a pharmacokinetics or interaction based on the dynamics. So how our drug interact one aspects, and how body respond to the particular drug, that is the another aspects. So how body respond to the drug, that is the kinetics of the drug. Kinetics can be easily identified with the movement of drug, how any medication is moving in the body through absorption, distribution, metabolism and excretion. And on the other hand, a dynamic is a mode of action of drug, how drug is targeting to the particular cell or maybe the receptors or maybe any enzymes. So if drug interact with a cell, it means we require a protein where drug can bind, and protein is serving as a carrier where drug is binding and reaching towards the target to show the desired pharmacological response. Age is also matters a lot, climatic factors matter a lot. In such cases, some genes are involved in such neurodegenerative and endocrinal disorders. There is one receptor involved for the Parkinsonism. So to address certain disorders based on the gene splicing or gene coding, in such cases, PINK1 type of genes has to be identified, and which are the biomarkers and cellular pockets are involved to address certain disease and disorders that acquires a lot of significant changes. Gender-based factors are there, disease-based factors are there, lifestyle modification are there. So overall, if we could figure out, so the point is very simple, how we are addressing certain lineage challenges to overcome genetic-based disorder, that is extremely important. Can we have the next? So here I would like to focus about the markers. What are those biomarkers? So biomarkers means where our drug is going to be interacted. So maybe any receptors, maybe the GPCR receptor, maybe the tyrosine kinase receptor, or maybe the response is based on the enzymatic association, or maybe any ion channel linked receptor, for example, sodium, potassium, calcium, chloride, how they are polarizing towards the cell, how they are depolarizing, and they are taking in and out to the particular drug to see the desired response. If you could see the endocrine system, so pituitary gland is the master gland who regulates the major hormones of the body. So hyperprolactin and prolactin, adrenocorticotropic hormones, growth hormones, luteinizing hormones, they are the endocrinological markers where we could identify and figure out the level of those hormones present in the plasma as well as serum to know the disturbance in the metabolic level. So it is also the metabolic disorder. There may be some comorbidities also associated with that. There may be the diabetes. Diabetes matters a lot because there are certain antipsychotic drugs which causes a development of diabetes as well, and later on it may lead to the severe neurodegenerative disorders like dementia. So those biomarkers are very precise, and through this artificial intelligence and molecular talking study, it is easy to figure out certain biomarkers to address neuroendocrine disorders. Next. So here I will discuss about the involvement of certain disease and disorders, how adrenal insufficiency can lead to the several disease and disorders, hyperthyroidism, diabetes, or myocarditis coma are there. So here if there are disturbance in the medulla and cortex, so we have to see what is the level of the adrenaline. Adrenaline is very important for the management of cardiovascular activity. Adrenaline manages the heart rate, pulse rate, cardiac rate. Also, it manages the blood pressure. If the level of the adrenaline is very high, so it may lead to a spontaneous expression over the blood pressure. So there may be chances of high blood pressure. If the level goes down, so it may lead to the cardiac activity. So certain behavior can be identified for the cardiovascular system, maybe the wakefulness kind of symptoms, maybe the fluctuation in the pulse rate or heart rate. So those type of symptoms are also very important to address certain neuroendocrinological disorders. If you could see the level of dopamine, so dopamine is one of the happiest hormones who is regulating certain many activities, which is responsible for cognition, orientation, focus, judgment, calmness, composedness of a healthy individuals. So those are the areas and aspects where we could address certain medical ambiguity which are not yet been addressed. There are several clinical trials are in the pipeline to identify certain disease targeted medication or disease oriented personalized medication to address certain disorder. Next. Here we are integrating immunology. Immune system is a very important fact to address any disease or disorders. So amyloid beta therapy for certain neurodegenerative disorders are in demand. Here in our India, if you could see, so AIMS Delhi is known for that. Now, some other private hospitals are also there. For example, Medanta Hospital, and if you could see, there is one more hospital in the Mumbai, that is the TCS, Tata Memorial Services. So they are the well-versed and advanced hospitals. They have certain immunological tools where we can employ amyloid beta therapy to enhance the immunity. Once the immunity develops, so it is an immunological response by the individual drugs. So once the immunological response is developed by any drugs, so there is no further administration of amyloid beta therapy is required, because it can be developed by the own antigen antibodies. Amyloid beta therapy is also available for the Alzheimer disease because there is a formation of plaque formation over the tangled protein because of the hyperphosphorylation. It delayed the response and it delayed the delivery of cellular signaling from body to the brain. Because brain has to receive the signal from the body, accordingly brain will address certain ambiguities and disorders, and it would release certain neurodegenerative disorders and the hormones like prolactin or thyroid stimulating hormones or the growth hormones for the development of brain, because it is responsible for the orientation and behavior. Altogether it is known as nootropic behavior or the cognitive behavior of any healthy individuals. Next. So, the overall gist of the presentation is that to address the neuroendocrinological disorders, we have to involve the personalized medication. In conclusion, we can say we have to increase our dependency towards the artificial tools and machine learning to capture the medical errors, or those data can be addressed in the pocket so that multiple adversity caused by the existing drug can be addressed. Thank you so much. I would like to convey my sincere gratitude and congratulations to the principal of Sitapur College. Also, I am very grateful for the invitation by the convener of this program. Also, I would like to express my sincere gratitude to Dr. Rohit Bargaji, who did a wonderful and magnificent job behind this conference. So, wishing you the great success. If any questions are there from the audience side, I would be happy to address. Thank you.
Any questions or any doubts from audience side? I think audience are very clear to your lecture, sir, and they have no doubts. Thank you so much, sir, for your complete session.
Thank you, madam. Thank
On
you.
behalf of our institution, Govt S.P.M College, Sitapur, and organizing committee, I extend my heartfelt gratitude to Professor Prasad Tiwari from from Sangha University, Bangalore. His presentation was masterfully navigated the complex intersection of nervous system and the endocrine system, showcasing the critical role of neuroendocrinology as a frontier in medicine. Your insights into the development of personalized medicine, especially how innovative approaches in drug delivery and compound research can tailor treatments for complex endocrine disorders. Thank you once again, Professor Tiwari. Thank you so much. For next technical session, we have with us Professor Pradeep Jamre, Department of Commerce, Government PG, Autonomous College, Durg. His topic is Business Management in Contemporary Times, Functions and Emerging Trends. Now, I request Professor Pradeep to come on the stage and deliver his valuable lecture. Welcome, sir.
Hello. Voice is clear?
कम कर दो, कम करो, बंद मत करो, कम करो।
गुड आफ्टरनून, बट मैं बोलना चाहूँगा गुड मॉर्निंग। गुड मॉर्निंग। अभी तक आप लोगों ने सुना था साइंस टेक्नोलॉजी, अब बारी है मैनेजमेंट। और मेरे ख्याल से ये 2 दिवसीय जो इंटरनेशनल कॉन्फ्रेंस चल रहा है उस पर मैनेजमेंट पर मेरा ही सिंगल टॉपिक है। क्लियर है? यहाँ पर मैनेजमेंट कॉमर्स के कितने स्टूडेंट हैं? कोई नहीं है? और साइंस बैकग्राउंड से और आर्ट्स से? वेरी गुड। चलो, क्योंकि मैं भी साइंस के स्टूडेंट रहा हूँ, बाद में कॉमर्स और मैनेजमेंट में गया, इसलिए साइंस टेक्नोलॉजी मैनेजमेंट एकदम रिलेवेंट टॉपिक है जिस पर आज इंटरनेशनल कॉन्फ्रेंस है। और मैनेजमेंट इंडस्ट्रियल रिवोल्यूशन के बाद यदि हम बात करते हैं तो मैनेजमेंट ही सबसे बेस्ट टॉपिक रहा है। तो मैं स्टार्ट करता हूँ अपने टॉपिक पे। सीतापुर कॉलेज के द्वारा 2 डेज इंटरनेशनल कॉन्फ्रेंस ऑर्गेनाइज किया जा रहा है, जिसका मेन जो टॉपिक है इमर्जिंग ट्रेंड इन साइंस टेक्नोलॉजी एंड मैनेजमेंट। और ऊपर सामने जो 2 पॉइंट दिख रहा है साइंस एंड टेक्नोलॉजी, वो दोनों पर भारी जो होने वाला जो पार्ट होता है वो मैनेजमेंट। यदि आप हिस्ट्री पढ़ेंगे मैनेजमेंट का तो जाकर के देखेंगे तो जो मैनेजमेंट रिवोल्यूशन हुआ है उन्हीं के बिहाफ में साइंस जनरेट हुआ। हम लोग हालांकि साइंस एंड टेक्नोलॉजी को मैनेजमेंट से आगे रखते हैं, बट मैनेजमेंट की जब बात आती है तो मैनेजमेंट रिवोल्यूशन सबसे टॉप में आता है। मेन पॉइंट पर मैं आना चाहूंगा। एक पिक्चर दिख रहा होगा, देखो, जो मैनेजमेंट के स्टूडेंट है शायद ये पिक्चर देखोगे, कॉमर्स मैनेजमेंट के स्टूडेंट पिक्चर देखोगे। एक एलिफेंट है और एलिफेंट को 5 लोग पकड़े हुए हैं, दिख रहा है पिक्चर? देखो, एक ने सूंड को पकड़ा हुआ है, एक कान को पकड़े हुए बैक साइड से, एक पैर को पकड़े, एक पूंछ को पकड़े, एक पेट को पकड़े, ऐसा कुछ पिक्चर दिख रहा है। आप लोगों को बोल के रखी थी फर्स्ट टेक्निकल पे, और आप लोग बात ही नहीं कर रहे। तो पिक्चर ही देखना है, बाकी कुछ नहीं करना है। सबसे छोटा प्रेजेंटेशन मेरा ही हो, इसलिए मेरे साथ रहो। यदि हम इसकी बात करते हैं, देखो जब मैनेजमेंट ओरिजिन हुआ, तो मैनेजमेंट ओरिजिन हुआ तो किसी के पास कुछ भी नहीं पता था। सब लोगों ने ये देखा कि ऐसा मैनेजमेंट हो। तो 5 ब्लाइंड पर्सन थे जिन्होंने क्या किया, मैनेजमेंट को इनोवेट किया। ओरिजिन जब ओरिजिन हुआ तो किसी ने कुछ को पकड़ा, उन्होंने बोला सांप टाइप का लग रहा है। किसी ने पैर को पकड़ा, उसको लगा कि बिजली का खंभा टाइप का लग रहा है। किसी ने पैर को पकड़ा तो बोला कि अरे यार ये इतना बड़ा है, कुछ भी समझ में नहीं आ रहा है। जिसने कान को पकड़ा, कान को पकड़ने वाले ने बोला कि सूट टाइप का लग रहा है। जिसने सूट को पकड़ा, उन्होंने बोला कि ये तो बांस की बल्ली होती है, बल्ली के बीच में गठान होता है, वैसा लग रहा है। तो मैनेजमेंट का कोई मैनेजमेंट का कोई फंडामेंटल प्रिंसिपल नहीं है। जैसे कि हम लोग साइंस में फंडामेंटल प्रिंसिपल पर जाकर के बात करते हैं, प्रूव करते हैं कोई चीजों को, वैसा मैनेजमेंट पर कोई ऐसा प्रिंसिपल नहीं। यदि आप ही के कॉलेज में पिछले बार सेमिनार हुआ होगा, उससे इस बार बेहतर हो रहा है, अगले बार और बेहतर होगा, किसका रोल है? मैनेजमेंट का। क्लियर आ गया? तो मैं मैनेजमेंट का डेफिनेशन या बहुत बड़ा लेक्चर देने वाला कुछ भी नहीं, क्लियर करके बताने वाला हूं कि जिस व्यक्ति ने जैसा देखा, जैसा सोचा, जैसा पाया, जैसा किया, वही मैनेजमेंट। आप जितना बेहतर कर सकते हो, बेहतर रिजल्ट ला सकते हो, वही मैनेजमेंट। और मैनेजमेंट पर जब हम लोग बात करते हैं तो यही सारे फंडामेंटल को आज के डेट में बड़े बड़े कारपोरेट यूज करते हैं। तो पहला पिक्चर क्लियर हो रहा है। सेकंड पिक्चर पर जब हम लोग बात किए तो दो लोगों में लड़ाई हुआ। एक इट इज साइंस, दूसरे ने बोला इट इज आर्ट्स। कॉमर्स तो था ही नहीं, मैनेजमेंट था ही नहीं। तो जब दो लोग के बीच में लड़ाई हुआ तो फिर फाइंडिंग किया गया कि भाई ये क्यों लड़ रहा है, ये क्यों लड़ रहा है। जब दोनों लोग लड़ रहे थे तो पता चला कि ये ना तो साइंस है ना तो आर्ट्स है। ये क्या है? साइंस आर्ट्स दोनों है। तो मैनेजमेंट अब केवल साइंस नहीं है और केवल आर्ट्स नहीं है, ये क्या है? बोथ है। नेक्स्ट स्लाइड में हम लोग बात करेंगे। अब हम लोग बात करते हैं कि फिर जब साइंस वालों की लड़ाई हुई तो साइंस पर एक ओरिजिन हुआ। साइंटिफिक मैनेजमेंट की बात करते हैं। जब साइंटिफिक मैनेजमेंट की बात करते हैं तो हम लोग एक व्यक्ति का नाम जरूर याद करते हैं। मैनेजमेंट के स्टूडेंट होंगे, उसको याद होगा, फ्रेडरिक टेलर। ही इज द फादर ऑफ साइंटिफिक मैनेजमेंट। इन्होंने कुछ प्रिंसिपल्स बताया जो साइंस से रिलेटेड है और साइंस से कम्युनिकेट करके उन्होंने प्रिंसिपल बनाया और उनके बेस पे इन्होंने इंडस्ट्री को आगे ले गए। क्योंकि हम लोग जब इंडस्ट्री रिवॉल्यूशन की अपन बात करते हैं तो कहां से बात करते हैं? यूरोप की इंडस्ट्री रिवॉल्यूशन की बात करते हैं। और ये अमेरिका का पर्सन था और यूरोप कंट्री पर इन्होंने साइंटिफिक इनोवेशन किया। तो सबसे पहले इन्होंने क्या किया? टाइम एंड मोशन स्टडी। साइंस बैकग्राउंड के विद्यार्थी होंगे वो पढ़े होंगे टाइम एंड मोशन। यदि हम इनकी बात करते हैं तो इन सभी चीजों को यूज किया और यूज करने से क्या हुआ? प्रोडक्टिविटी बढ़ी। मतलब हर चीज का एक सिस्टमैटिक टाइम यूज किया। दूसरा लेबर, ठीक है, लेबर को भी साइंटिफिक वे पर काम डिवीजन किया, उनके बेस पे प्रोडक्शन अपने आप बढ़ना था। इनका एग्जांपल अपने इंडियन कॉन्सेप्ट पर लेकर अगर चलते हैं तो टाटा स्टील ने सबसे पहले साइंटिफिक मैनेजमेंट यूज किया। टाटा स्टील ने साइंटिफिक मैनेजमेंट यूज किया और इनको वर्ल्ड लेवल पर मोस्ट एथिकल कंपनी का अवार्ड मिला। दूसरा चीज है, कोई भी चीजों को अपन को जनरेट करना होता है तो एक स्टैंडर्ड होना चाहिए या एक प्रोसेस होना चाहिए। क्योंकि साइंस की हम लोग बात करते हैं, साइंस कोई भी चीज को जब प्रूव करता है तो स्टेप बाय स्टेप प्रूव करता है और देन फाइंडिंग पे जाता है, रिजल्ट निकलता है। तो यहां पर जब हम लोग बात कर रहे हैं, हमने फॉर एग्जांपल लिया है बैंक। बैंक पर जब कोई भी काम करने जाओगे तो आपको रो पे लगना ही पड़ता है, प्रॉपर वे पे काम करना पड़ता है। आपको उसको बैंक के रूल्स को, स्टैंडर्ड को फॉलो करना पड़ता है। जैसे कि फॉर एग्जांपल यहां पर दिया गया है, भाई जब आप बैंक जाते हैं, बैंक में लोन लेने जाते हैं तो लोन लेने जाएंगे तो सबसे पहले आप लोन के लिए एलिजिबल है या नहीं एलिजिबल है उसके लिए क्या किया जाता है? डॉक्यूमेंट को वेरिफिकेशन किया जाता है। फिर आपकी डेटा एंट्री की जाती है, उसके बाद आपका क्रेडिट स्कोर या सिविल स्कोर चेक किया जाता है, फिर आपका लोन अप्रूवल होगा। तो ये सारे प्रोसेस मैनेजमेंट के थ्रू यहां पर आए। नेक्स्ट हम लोग बात कर रहे हैं मैनेजमेंट की बात कर रहे हैं, लेबर की और मनी की अपन बात कर रहे हैं। यदि जितने भी आईआईटीएन बच्चे, यदि आईआईटीएन कोई किसी पर्सन से मिलोगा तो उनका ड्रीम जॉब होता है यदि गूगल में हमको जॉब मिले। यानी हम इंडिया की बात कर रहे हैं या पूरे वर्ल्ड की बात करें तो जितने भी आईआईटीएन है, वर्ल्ड के टॉप जो आईआईटीएन होते हैं वो कहां पर जॉब सर्च करना चाहते हैं? गूगल। क्योंकि गूगल पे इसलिए आप सर्च करना चाहते हैं क्योंकि वहां पर हर प्रकार के काम आपके अपने अनुसार काम करें, आपको किसी प्रकार का कोई बंधन नहीं, ठीक है, किसी प्रकार का कोई पनिशमेंट नहीं। और ये सब के कारण क्या है, ये सब के कारण क्या है, वो टॉप की कंपनी। अब सब लोग अवेयर है गूगल के नाम से, कुछ बताने की जरूरत ही नहीं है, गूगल कंपनी इतनी बड़ी कंपनी है। उसके बाद हम लोग बात करते हैं ट्रेनिंग एंड डेवलपमेंट। जब ट्रेनिंग एंड डेवलपमेंट की अपन बात करते हैं तो टोयोटा कंपनी का कार आप लोगों ने देखा होगा, इनोवा वगैरह नाम सुने होंगे, इनोवा कार का नाम सुने होंगे आप लोग, पीछे वाले सुन ही नहीं रहे हैं, सुनो। तो यहाँ पर आज एचडीएफसी बैंक का नाम सुने आप लोग, सीतापुर में बहुत सारे बैंक हैं, एचडीएफसी है कि नहीं है? यदि एचडीएफसी बैंक में आपको कॉलेज में काम करना होगा तो उसके लिए ऐसा जरूरी नहीं है कि मैनेजमेंट स्टूडेंट हो, कॉमर्स स्टूडेंट हो, आर्ट्स स्टूडेंट हो, साइंस स्टूडेंट हो। आज के डेट में एचडीएफसी बैंक में किसी को भी रिक्रूटमेंट किया जाता है तो सबसे पहले आपकी काउंसलिंग करता है, काउंसलिंग करने के बाद क्या करेगा, आपको 3 महीने की ट्रेनिंग देगा, 3 महीने की ट्रेनिंग के बाद ही आपको क्या करेगा, वो अपने जॉब पे रखेगा। तो आज के डेट में जितनी भी कंपनी है, साइंटिफिक मैनेजमेंट में हम लोग इस पार्ट को इसलिए रखे हैं क्योंकि सारी कंपनी अपने आप को ग्रोथ चाहती है, क्योंकि वो ट्रेनिंग देने के बाद उनका टेस्ट लिया जाता है, टेस्ट लेने के बाद ये किया जाता है कि वो हमारे लिए एलिजिबल है कि नहीं, और जब एलिजिबल हो जाता है तभी उनको जॉब ऑफर किया जाता है ताकि वो अपनी कंपनी पर करेक्ट रोल अप्रूवल दे सके। अब हम लोग बात करते हैं, मैंने कुछ ही पॉइंट को लिया था साइंटिफिक मैनेजमेंट का जो बेसिकली यूज होता है। अब दूसरा आर्ट्स ग्रुप में हम लोग बात करते हैं एडमिनिस्ट्रेटिव मैनेजमेंट की, अपन जब बात करते हैं तो यहाँ पर एक पर्सन का नाम आता है हेनरी फेरो। ये फ्रांस का पर्सन था और इन्होंने 14 प्रिंसिपल दी। यदि कल में आप लोग सिविल सर्विस की एग्जाम की तैयारी करेंगे तो 14 प्रिंसिपल आप हेनरी फेरो जरूर पढ़ेंगे। और पब्लिक एडमिनिस्ट्रेशन यदि पढ़ना होगा तो वहां पर इनका प्रिंसिपल आपको जरूर पढ़ना। तो इनके प्रिंसिपल को हम कोऑर्डिनेट करेंगे किसके बेस पे, कंपनी के बेस पे। 14 प्रिंसिपल मैं यहाँ पर दिखा रहा हूँ। अब इनकी जो प्रिंसिपल है, सबसे पहले इन्होंने प्रिंसिपल जो दिया डिवीजन ऑफ वर्क, कि कौन व्यक्ति को किस किस प्रकार के काम दिया जाए। जैसे कि यहाँ पर काम दिया गया है, एक को फोटोग्राफी करने का काम दिया गया है, ठीक है, एक को खाना अरेंजमेंट करने का काम दिया है, एक को टेक्निकल अरेंजमेंट करने का काम दिया गया है। तो यहाँ पर जितनी भी प्रकार के लोगों की क्वालिटी को देख करके काम दिया गया है। तो वैसे कॉर्पोरेट कल्चर में भी ये डिवाइड हो गया है कि किन पर्सन से किस प्रकार का काम लिया जाए। पॉइंट नंबर 2, अथॉरिटी एंड रिस्पांसिबिलिटी। ऐसा नहीं हमेशा हम लोग क्या करते हैं, अपने अधिकार के लिए लड़ते हैं और लायबिलिटी के लिए क्या करते हैं, भगते हैं। तो सेकंड प्रिंसिपल पे हमको ये बताया गया है मॉडर्न मैनेजमेंट में कि आपकी अधिकार तो है, अधिकार के साथ साथ क्या है आपकी जिम्मेदारी, और जिम्मेदारी को भी आपको क्या करना पड़ेगा, निर्वहन करना पड़ेगा। नेक्स्ट है डिसिप्लिन, क्योंकि जैसे कि आज हम लोग डिसिप्लिन हो करके बैठे हैं या कुछ लोग जबरदस्ती बैठे, दो ही प्रकार के लोग होंगे। कुछ लोग कुछ सीखेंगे करके बैठे होंगे और कुछ लोग चलो यार 2 दिन झेल लेते हैं करके बैठे होंगे। कितने लोग ऐसा हाथ ऊपर कर दो थोड़ा सा, नो है ना? तो चलो फिर इसकी अपन बात करते हैं, क्योंकि इसके बगैर आप कहीं पर भी सक्सेस नहीं हो सकते। ना मैं साइंस की बात करूं, ना आर्ट्स की बात करूं, ना मैनेजमेंट की बात करूं, बगैर डिसिप्लिन के आप किसी भी जगहों पर काम नहीं कर सकते। और मार्केट में हर कंपनी पर, क्योंकि आने वाले समय में गवर्नमेंट सेक्टर भूल जाओ, कॉर्पोरेट कल्चर ही होने वाला है 2030 के बाद। और कॉर्पोरेट कल्चर में आपको सर्वाइव करना है तो डिसिप्लिन, क्योंकि आज के जनरेशन के जो स्टूडेंट्स हैं उन पे किसी प्रकार की डिसिप्लिन है ही। नेक्स्ट पॉइंट पर अपन बात करते हैं यूनिटी ऑफ कमांड, क्योंकि बड़ी जो कॉर्पोरेट इंस्टीट्यूट होते हैं वहां पर क्या होता है, जो टॉप लेवल का जो ऑफिसर होता है वो कमांड देता है और लोअर लेवल के ऑफिसर तक वो सर्वाइव एक लाइन पर चलता है और उनके कामों को क्या करना होता है, फॉलो करना। नेक्स्ट पॉइंट है यूनिटी ऑफ डायरेक्शन, क्योंकि हम टारगेट पहले फिक्स करते हैं, उसके बाद क्या करते हैं, प्लानिंग करते हैं। क्योंकि मैनेजमेंट का पहला प्रिंसिपल जो होता है वो क्या होता है, प्लानिंग। बगैर प्लानिंग के हम कोई भी चीजों पर सक्सेस नहीं हो सकते। यदि आज इंटरनेशनल कॉन्फ्रेंस ऑर्गेनाइज हुआ है तो 2 महीना पहले का प्लानिंग चल रहा था, तब आज हम लोग यहाँ पर खड़े हैं। नेक्स्ट पॉइंट है सबऑर्डिनेट ऑफ इंडिविजुअल इंटरेस्ट टू ऑर्गेनाइजेशनल इंटरेस्ट। यहाँ पर जब बात करते हैं, देखो हिंदी का जो वर्ड है इतना अच्छा वर्ड है, संगठन ही सर्वोपरि। तो यदि मैं यहाँ के विद्यार्थियों की बात करूं तो मेरा भी महाविद्यालय सर्वोपरि, क्लियर है, मेरा भी महाविद्यालय सर्वोपरि। जैसे कि मैडम बताइए कि कहीं पर गुटखा का पाउच भी दिख गया तो आपको उठा करके डस्टबिन पे डाल देते। तो यदि आप ऐसा नहीं करेंगे तो यही सेम कॉन्सेप्ट कहां पर लागू होता है, ऑर्गेनाइजेशन पर लागू होता है। नेक्स्ट पॉइंट है रेमुनेशन। अब हम कॉर्पोरेट कारपोरेट की बात करें, कंपनी की बात करें, तो वहां पर जिनका जिस प्रकार का काम है उस काम के बिहाफ में क्या मिल रहा है उनको, रेमुनरेशन मानदेय मिल रहा है। नेक्स्ट है फेडरलाइजेशन एंड डिफेडरलाइजेशन। यहाँ पर दोनों प्रकार की कम्युनिकेशन की अपन जब बात कर रहे हैं या वर्क की बात कर रहे हैं, तो कई बार ऐसा होता है कि जो टॉप लेवल ऑफिसर वही हमको काम दिया उन्हीं को हम कर रहे हैं और उन्हीं के अकॉर्डिंग काम कर रहे हैं। कई बार हमारा क्या लोअर लेवल का ऑफिसर होता है, उनकी बातों को ऊपर सुनी नहीं जाती। तो इसलिए मैनेजमेंट पर ये लाया गया कि हम दोनों प्रकार के लोगों को फेडरलाइज और डिफेडरलाइज दोनों वे पर होकर के काम करें। आप लोग बेटा बैठ जाओ पहले। नेक्स्ट है स्केलर चेक। कोई भी ऑर्डर है एक मैनेजर से लेकर के, जनरल मैनेजर से लेकर के लोअर लेवल के सारे सब ऑफिसर को धीरे धीरे आता है, एक चेन के बेस पे आता है। तो ऑर्डर की हम जब बात करते हैं, इसका एक एग्जांपल बताऊंगा। जैसे कि फॉर एग्जांपल में हम लोगों ने लिखा है हर वस्तु या सही व्यक्ति को सही स्थान मिले, तो सभी चीजों के लिए प्रॉपर प्लेस होना चाहिए। उनको अपन ऑर्डर पर समझेंगे आगे जाकर के। इक्विटी, यदि एक समान लेवल का काम है तो समान वेतन भी मिलना चाहिए, समान समय पर प्रोडक्टिविटी होनी चाहिए। जब हम किसी कारपोरेट की बात करते हैं तो वहां पर सभी को एक बराबर एक ही तराजू पर तोला जाता है और एक ही प्रकार के प्रोडक्शन की बात की जाती है। स्टेबिलिटी ऑफ टेलर, इसकी अपन बात कर रहे हैं। आज की डेट में लोग जॉब करने जाते हैं, प्राइवेट सेक्टर में जॉब करते हैं, 15 दिन करते हैं, 1 महीना करते हैं, छोड़ के आ जाते हैं। क्योंकि हमारे बच्चे, हमने बहुत सारे अपने कॉलेज में बहुत सारी कंपनियों को ठीक है प्लेसमेंट के लिए बुलवाया था। हमारे बच्चे कुछ दिन ट्रेनिंग लेकर के गए, फिर 2 महीना 3 महीना जॉब किए, फिर छोड़ के आ गए। तो इसलिए आज के डेट में पहले आपको उस कंपनी के लेवल के लोग बना दिया जाता है, उसके बाद क्या किया जाता है, आपको वहां पर रखा जाता है ताकि आप उस कंपनी को छोड़ के न जाए। छोड़ के जाएंगे तो क्या होगा, कंपनी को बार बार रिक्रूटमेंट करना पड़ेगा और बार बार रिक्रूटमेंट करने से अनट्रेंड लोग आएंगे और अनट्रेंड लोग आएंगे तो कंपनी को क्या होगा, घाटा होगा। नेक्स्ट प्रिंसिपल पर बात कर रहे हैं, इनिशिएटिव। यहाँ पर ओपन कल्चर, ओपन कम्युनिकेशन कल्चर की हम बात कर रहे हैं। कर्मचारियों के सुझाव देने की स्वतंत्रता है, मतलब जितने भी एम्प्लॉय हैं वो टॉप मैनेजर या कंपनी के जो ओनर है उनको भी अपना सजेशन दे सकते हैं। और ये चीज आप लोग सुने होंगे, रतन टाटा जी ऐसा करवाते थे। ये एग्जाम्पल आप लोग सुने होंगे कि रतन टाटा जी इस प्रकार का काम करवाते थे। हर छोटे छोटे एम्प्लॉय की बात करते थे, उनके साथ भी बैठ के चाय पीते थे। अचानक छोटे किसी को बुला लेते थे कि इन एम्प्लॉय को भेजो, उनके साथ मैं बैठ के चाय पीऊं। नेक्स्ट है स्पिरिट डेकोर। और कोई भी कारपोरेट की हम जब बात करते हैं तो वहां पर बगैर टीम वर्क के कोई भी चीज सक्सेस नहीं हो सकती। जैसे हमने स्टार्टिंग में यहाँ पर बोला कि ये जो पूरा कॉन्फ्रेंस चल रहा है, ऑर्गेनाइज चल रहा है, ये पूरी टीम करोगे। अकेले प्रिंसिपल कुछ नहीं कर सकता, अकेले कन्वीनर कुछ नहीं कर सकता, पूरी टीम, पूरे स्टूडेंट से लेकर के हमारे क्लास 3 क्लास 4 के एम्प्लॉय है, उन सबका रोल होगा, तब ये चीज हो रहा है, इसलिए ये चीज सक्सेस हो रही है। तो कारपोरेट में भी क्या होता है, इस प्रकार की टीम कल्चर डेवलप की जाती है ताकि वह कंपनी आगे बढ़ सके। हमने कुछ एग्जांपल लिया है, जैसे कि डिवीजन ऑफ वर्क पर काम किया किसने, मारुति सुजुकी। और मारुति सुजुकी आज हर व्यक्ति जानता है कि मारुति सुजुकी क्या है। स्टूडेंट आप लोग जानते हैं कि मारुति सुजुकी क्या है, टैबलेट बनाने वाली कंपनी ना, टैबलेट बनाती क्या, मारुति सुजुकी एक कंपनी है, हमने नाम लिखा है, वो क्या बनाता है, कुछ बनाता है, 2 व्हीलर 4 व्हीलर 4 व्हीलर 2 व्हीलर नहीं बनता, बनता है, ठीक है। तो उन्होंने ये प्रयोग किया, यानी मॉडर्न प्रिंसिपल का यूज किया। दूसरा यूनिटी ऑफ कमांड। यहां पर हम लोग एक एग्जांपल लिए इंडियन रेलवे का। इंडियन रेलवे देखो, मतलब इंडिया की अपन बात करें तो सबसे ज्यादा एम्प्लॉय जनरेट करने वाली कंपनी है इंडियन रेलवे। और इंडियन रेलवे पे यूनिटी ऑफ कमांड, एक जो सीनियर ऑफिसर जो बोलता है भाई, क्योंकि वहां पर ऐसा नहीं है कि जूनियर का बोला हुआ चीज को मान लिया तो पता नहीं सिग्नल फेल होगा, कब ट्रेन एक्सीडेंट। इसलिए क्या है, वहां पर जो टॉप लेवल ऑफिसर होता है, जो सीनियर ऑफिसर होता है, उन्हीं की कमांड के बेस पे ऑर्डर निकलता है। नेक्स्ट पॉइंट हम लोग बात करें स्केलर चेन। रिलायंस इंडस्ट्री, जियो का सिम वगैरह इस प्रकार की चीज सुने होंगे आप लोग, सबके पास होगा, मोबाइल तो होगा सबके पास, तो जियो का सिम भी होगा। और रिलायंस यानी अंबानी फैमिली का जितना भी काम होता है रिलायंस इंडस्ट्री के अंदर होता है। ऐसा नहीं होता है जियो एक पार्ट है उसका, जियो एक छोटा सा पार्ट है ना, बहुत बड़ा पार्ट नहीं है, उससे भी और बड़ा बड़ा काम होगा। तो तो यहाँ पर वो क्या करता है, जितने भी वर्कर है उनका एक चैनल बनाया कि ये काम ये करने के बाद इनके पास जाएगा, उसके बाद इसके पास जाएगा, उसके बाद इसके पास जाएगा। तो एक चैन सिस्टम बेस पे काम है और चैन सिस्टम से काम करने पर वो कंपनी ग्रोथ। यदि हम लोग यहीं पर बात करते हैं, धीरू भाई अंबानी के दो बेटे मुकेश और अनिल, मुकेश और अनिल, और आज के डेट में हम लोग नाम लेते हैं तो केवल किसकी नाम लेते हैं, मुकेश की कि अनिल का? मुकेश का, क्योंकि उनकी कंपनी पर भी हम लोग बात नहीं कर रहे हैं। आगे मैं बढ़ रहा हूं थोड़ा सा। एक टाटा ग्रुप के लिए है, यदि टाटा ग्रुप में आप नौकरी कर रहे हैं और अभी हाल ही में कोविड पेंडेमिक हुआ था, तो टाटा ऐसा कंपनी था जिन्होंने किसी को नौकरी से निकाला, टीसीएस। बाकी सारी कंपनी ने क्या किया, छटनी कर दिया, लोगों को निकाल दिए। लेकिन इनकी जब कंपनी की बात करते हैं तो आप इंडिया में जब किसी को जॉब चाहिए तो लोग बोलते हैं टीसीएस में कर लें। तो टीसीएस एक अच्छी कंपनी है जिसपे कर्मचारियों का मतलब गवर्नमेंट एम्प्लॉय की तरह सारी फैसिलिटीज प्रोवाइड होता है। उसके बाद हम लोग बात कर रहे हैं, इन्फोसिस कंपनी का नाम सुने होंगे, नहीं सुने? सुने। कॉमर्स विद्यार्थी बड़े सब पर केस स्टडी करना है, ठीक है। मैं फरीदा मैडम को भी बता दे रहा हूँ, आप सभी बच्चों को केस स्टडी करना है एक एक पॉइंट पे। एक टाटा कंपनी का लेकर के केस स्टडी कर लेना, एक इन्फोसिस लेकर के कर लेना, एक एचडीएफसी पे कर लेना, क्योंकि मैडम ने भी थोड़ी देर पहले बोल के गई आपको एनईपी आना चाहिए। और एनईपी आना चाहिए मतलब आपको सभी को सभी विषय आना चाहिए, ऐसा नहीं है। इसलिए हम लोग बात नहीं कर रहे हैं ये कॉमर्स है कि मैनेजमेंट है कि आर्ट्स है कि टेक्नोलॉजी, सभी एक प्लेटफॉर्म पे। उसके बाद एक ऑर्डर की एचडीएफसी की मैं बात कर लिया था, ऑर्डर की बात कर रहे हैं। अमेजन से हम लोग सब कोई क्या करते हैं, ऑर्डर करते हैं। अमेजन, फ्लिपकार्ट, मीशो, लड़कियों तो एकदम। और ये कहां तक पहुंच गया, गांव तक पहुंच गया। देखो सबको यस सर पहुंच गया ना गांव तक। और ये कहां की कंपनी है नहीं पता, कहां की कंपनी है, छत्तीसगढ़ की, भारत की, सब लोग भारत की बोलते हैं। अमेजन के जंगल का नाम सुने हो, कहाँ है, भारत में, मेन पार्ट में। अमेरिका की कंपनी भाई। और यहाँ पर जब आप कोई भी चीज को ऑर्डर देते हैं तो ऑर्डर डिस्पैच, फिर दिल्ली से निकला यदि कोई प्रोडक्ट है तो कहाँ पहुंचा, नागपुर पहुंचा, नागपुर के बाद रायपुर पहुंचा, रायपुर के बाद सरगुजा पहुंचा, सरगुजा के बाद हमारा सीतापुर पहुंचा, फिर आउट ऑफ फॉर डिलीवरी। ऐसा आप लोग देखें कि नहीं देखें? और इनकी सारी सिस्टम किसके बेस पे हो रही है, रोबोटिक्स पे हो रही है। इनके जो गोडाउन है वहां पर पूरा रोबोट काम करते हैं। यदि आप एक आध दो रील जब देखते हैं ना इंस्टा पे, तो इस प्रकार का भी रील देखा करें। आप लोग सब रील देखते हो, स्नैपचैट सब चलाते हो, लेकिन ये चीज भी देख लो उसी रील पे तो ज्यादा बेहतर होगा। क्लियर है? तो ये कुछ एग्जांपल्स थे किसके, मॉडर्न मैनेजमेंट के। तो अब हम लोग लड़ाई जो कर रहे थे साइंस और आर्ट्स की, वो चीज खत्म। मेन प्रिंसिपल पर हम बात करते हैं, जब फंक्शन ऑफ मैनेजमेंट की बात करते हैं, सबसे पहले आता है प्लानिंग, ऑर्गेनाइजेशन, कोऑर्डिनेशन, लीडरशिप, कंट्रोलिंग, कम्युनिकेशन, इस प्रकार की चीजें आती हैं। अब ये सारी चीजों को कोऑर्डिनेट कैसे करना मैं आगे बता रहा हूँ। आगे जब बता रहा हूँ तो एक पिक्चर दिख रहा है, देखो पिक्चर दिख रहा है किसका, कुछ लड़ाई कर रहे हैं, इस प्रकार का दिख रहा है। इसका मतलब मैं यहाँ पर महाभारत करवाने के लिए खड़ा नहीं हूँ। अब पूरे वर्ल्ड पे जब अभी तक हम लोगों ने बात किया था, अमेरिका की बात किया था, फ्रांस की बात किया था, लेकिन इंडियन मैनेजमेंट सिस्टम जो हमारा भगवत गीता पे दिया है, जब महाभारत का युद्ध हुआ उस दौरान पे भगवान कृष्ण ने अर्जुन को जो उपदेश दिए, और वो बुक हम लोग को पढ़ना चाहिए। लेकिन फॉरेन कंट्री में कॉस्मोस नामक की बुक के थ्रू ये पूरे वर्ल्ड के मैनेजमेंट स्कूल है, वहां पर पढ़ाया जाता है, और हमारे बच्चे हमारे यहाँ की चीज को नहीं पढ़ते। क्लियर है? तो आप लोगों को एक बुक पढ़ना है, कौन सा, कॉस्मोस, और उसको नहीं पढ़ सकते तो चुपचाप भगवत गीता पढ़ लो। और दुनिया का सबसे बेस्ट जो मैनेजमेंट है भगवत गीता। क्लियर है? अब मैं उनको प्रूव कर रहा हूं आगे। सबसे पहले एक फोटो देखना है, बाकी ऊपर क्या है उसको कुछ नहीं देखना। एक आप देखो, अर्जुन है, द्रोणाचार्य है, और ऊपर पेड़ पे पक्षी दिख रहा है, और ये वाला कहानी आप सब लोगों ने सुना होगा। अब देखो हमने टाइटल डाला है गोल एंड क्लियर विजन, और ये जो अर्जुन का जो पिक्चर है उस पिक्चर को कॉपी करके कौन आगे बढ़ गया? बगल में एक और इमेज दिख रहा है, देखो किसका दिख रहा है? गूगल का। सब लोग यूज कर रहे हैं गूगल का। और वो कहां से लिया? क्योंकि गूगल एक ऐसी कंपनी है जिनका एकदम विजन क्लियर है, एकदम क्लियर विजन पे काम करता है। और क्लियर विजन पे काम करने के कारण और कहां से सीखा पता है? वो हमारा जो पहला जो प्रिंसिपल अपन बताया ना, बिना लक्ष्य के कोई भी संगठन सफल नहीं हो सकता, ये टैग लाइन भगवत गीता से किसने लिया? गूगल ने लिया। तो पहला पिक्चर पर अब कुछ नहीं बोलूंगा, क्लियर हो गया सबको? तो आपको समझना है इस चीज। सेकंड पॉइंट भी दिख रहा है कुछ, टीम वर्क। अब यहां पर अपन बात कर रहे हैं राइट मैन फॉर द राइट जॉब। यदि आप भगवत गीता पर बात करते हैं या महाभारत पर बात करते हैं तो वहां पर जिनकी टीम अच्छी थी, जिनकी टीम मतलब सही व्यक्ति को सही काम मिला, वो टीम विजेता हो। ऐसा ही हुआ। जैसे कि आज की हम लोग बात करते हैं तो यहाँ पर इस कॉलेज में जो कॉन्फ्रेंस हो रहा है उनकी पूरी टीम और पूरे टीम कोऑर्डिनेट होकर के काम कर रहे हैं, हम सफल हो जाए। तो यहाँ पर जब हम लोग बात कर रहे हैं, यदि चाहता तो भगवान कृष्ण भी क्या कर सकता था? युद्ध कर सकता था, लेकिन किया क्या? नहीं किया। उन पर हम आगे बात करेंगे उन पर थोड़ी सी। अब यहाँ पर हम जब बात कर रहे हैं तो यहाँ पर लोगों भगवान कृष्ण ने क्या बन गया? भगवान कृष्ण जब युद्ध के बात आई, महाभारत होगा करके डिक्लेयर हो गया, तो दो लोग भगवान कृष्ण के पास गए। कौन कौन? दो लोग। एक तो गया कौन? दुर्योधन। दूसरा कौन गया? अर्जुन। और दोनों में सबसे पहले कौन गया? दुर्योधन। और जब दुर्योधन गया तो भगवान कृष्ण क्या कर रहे थे? सो रहे थे। जब भगवान कृष्ण सो रहे थे तो दुर्योधन क्या किया? भगवान कृष्ण के सिर के पास जाके बैठ गए। थोड़ी देर बाद क्या पहुंचा? अर्जुन पहुंच गए वहां पे। अर्जुन उनके पैर की तरफ बैठ गए। जब भगवान कृष्ण सोकर उठे तो उन्होंने बोला सबसे पहले किसको देखे? दुर्योधन की या अर्जुन को? अर्जुन को। तो उन्होंने तुरंत आंख खुलते ही पूछा कि अर्जुन कैसे आए? तो बोले कि भाई आपको हमारे साथ रहना है। तो दुर्योधन बोलने लगा कि मैं पहले आया, मेरे साथ रहोगे आप। दुर्योधन बोला कि मैं पहले आया, मेरे साथ पहले रहो। तो भगवान कृष्ण ने बोला कि भाई मेरी नजर जिन पे पहले पड़ी है उनके साथ। तो दुर्योधन फिर वाद पे आ गए। तो भगवान कृष्ण ने क्या किया? दोनों को बोला कि एक तरफ मैं बिना अस्त्र शस्त्र के और दूसरी तरफ मेरी पूरी नारायणी सेना। अब इनका सिंपल एग्जांपल ले रहा हूं। जैसे मैं इन्हीं की बात कर रहा हूं, भगवान कृष्ण की बात कर रहा हूं। आज के डेट में अपन बात करें, हम लोग यहां पर हैं। यदि अमेरिका में डोनाल्ड ट्रंप को लेना चाहोगे कि अमेरिका की पावर या एयरफोर्स मिसाइल ये सब लेना चाहोगे? आज मैं बात कर रहा हूं यदि आपको अमेरिका की पूरी सेना को चूज करना हो और दूसरा डोनाल्ड ट्रंप को चूज करना हो, किसको चूज करो? सेना को। क्योंकि अभी हमारा विजन क्लियर है, एकदम क्लियर, इसलिए हम सेना को चूज करेंगे। और लेकिन उस समय किसी को कुछ भी नहीं पता था, इवन तक अर्जुन को भी नहीं पता था कि मैं भगवान को रखूं। लेकिन दुर्योधन ने क्या किया? हम भाई नारायणी सेना रखेंगे, सेना इतना हमारे स्ट्रांग सेना हो जाएगा। ये लोग तो 5 लोग हैं और अर्जुन मिलाकर के 6 लोग होंगे, आराम से लड़ाई जीत लेंगे। यहाँ पर इन्हीं की मैं बात कर रहा हूँ, उन्हीं लीडरशिप की बात कर रहा हूँ। सैनिक नहीं थे फिर भी इनको यूज किया किसने? रतन टाटा ने अपनी कंपनी पर यूज किया। कम्युनिकेशन की अपन बात कर रहे हैं। जब अर्जुन कुछ दिनों बाद, जब लड़ाई कितने दिन तक चली थी? चलो क्वेश्चन नहीं पूछ रहा हूं मैं। एक समय के बाद अर्जुन को लगा कि मेरे पूरे परिवार के लोग हैं जिसको मैं मार रहा हूं, वो अस्त्र छोड़ दिए थे। तब भगवान कृष्ण ने उनको पूरा उपदेश दिया और ये उपदेश पूरा इफेक्टिव कम्युनिकेशन कराता है। तो आप लोग इस चीजों को थोड़ा सा पढ़िएगा। अब ये स्ट्रेटजी एंड इनोवेशन, जैसे कि उस समय पर इनोवेशन किया गया था कि भीष्म पितामह को कोई हरा नहीं सकता था, तो सिनेटी का यूज करके एक स्ट्रेटजी बनाया गया। क्लियर है? और इसी चीज को स्ट्रेटजी स्टीव जॉब्स ने अपनी कंपनी पर किया। इस पर एप्पल का मोबाइल आप लोग सुने होंगे, यूज भी कर रहे होंगे। उन्होंने आईफोन पर यूज किया, आईट्यून पर यूज किया, आईपैड पर यूज किया, मैकबुक पर यूज किया। तो कारपोरेट पूरा भगवत गीता का जो मैनेजमेंट प्रिंसिपल है उसके बेस पे चल रहा है, और हम नहीं यूज कर रहे हैं, फॉरेन कंट्री यूज कर रहे हैं। ये मारुति सुजुकी वाला कहानी लगभग मैं बता चुका था। अब हम लोग कुछ मैनेजमेंट के ऊपर बात करते हैं। आज के डेट में हमको जो बड़ी जो कॉर्पोरेट कंपनी है उनके समय पर इस प्रकार की चीज होना चाहिए। तो पहला पॉइंट है डिजिटल ट्रांसफॉर्मेशन मैनेजमेंट। अब ये मैनेजमेंट का नया प्रिंसिपल, क्योंकि आज के डेट में जब डिजिटल नहीं होंगे, जब तक डिजिटल नहीं होंगे, सारी कंपनी डिजिटल नहीं होगा तक एक समय में बैंक हुआ, बैंक के बाद कोऑपरेटिव बैंक भी आज के डेट में डिजिटल हो गए। तो डिजिटल नहीं होंगे तो आप आगे नहीं बढ़ पाएंगे। क्लियर है? और डिजिटल में सबसे ज्यादा रोल किसने अदा किया? रिलायंस और जियो। नेक्स्ट पॉइंट पे कस्टमर सेंट्रिक मैनेजमेंट। अमेजन की फिर बात है, वही आ रही है अमेजन कंपनी की। आज के डेट में सारी जितनी भी कंपनी अपना प्रोडक्ट कंज्यूमर बेस्ड प्रोडक्ट बना रही है, और इनके लिए उन्होंने एक अलग मैनेजमेंट कस्टमर सेंट्रिक मैनेजमेंट सिस्टम लागू कर दिया है, क्योंकि वे गांव पे गांव के ग्राहक को भी क्या कर रहे हैं? खोज रहे हैं। और जिसका मैंने एग्जांपल अभी दिया था अमेजन, आपके गांव तक पहुंच गया। एक गांव के छोटा भी कस्टमर है उनको भी फोकस कर रहे हैं। इनोवेशन ड्रिवन मैनेजमेंट। पहले हम लोग किस पर ट्रांजैक्शन करते थे? रोकड़ पे ट्रांजैक्शन करते थे, किस पे ट्रांजैक्शन करते थे? आज एक छोटा सा ठेला वाला, गुपचुप वाला, जिसके पास भी क्या होता है? यूपीआई होता है। क्लियर है? तो अब मैनेजमेंट का सिस्टम क्या हो गया? बदलते जा रहे हैं। इनोवेटिव जब तक नहीं होंगे तब तक आप कुछ नहीं कर पाएंगे। ह्यूमन वैल्यू मैनेजमेंट। एक समय पे हम लोग मैनेजमेंट में पढ़ाते थे, ह्यूमन रिसोर्स मैनेजमेंट पढ़ाते थे। आज डेट में ह्यूमन वैल्यू मैनेजमेंट हो गया, जिसकी कंपनी पर ठीक है एम्प्लॉय हैप्पी होगा, हैप्पी एम्प्लॉय होगा तो हायर प्रोडक्टिविटी होगी, लॉयल्टी होगी, फिर क्रिएटिविटी। तो एक पर लोगों की वैल्यूज के बेस पे क्या हो रहा है? आज डेट में मैनेजमेंट क्रिएट हो जा रहा है। तो ह्यूमन वैल्यू मैनेजमेंट आज डेट में एचआरएम की जगह ले चुका है। सस्टेनेबल एंड ग्रीन मैनेजमेंट, मैडम का भी टॉपिक था इस पे, जिसपे वेस्ट मैनेजमेंट। तो आज के डेट में कंपनी भी क्या है? वेस्ट मैनेजमेंट बेस पर काम करके प्रोडक्ट दिए, मतलब ग्रीन प्रोडक्ट। जैसे कि आप लोग ग्रीन एनवायरनमेंट पर अभी बात कर रहे थे, ग्रीन यूनिफॉर्म पर बात कर रहे थे, वैसे ही आज में कारपोरेट क्या कर रहे हैं? ग्रीन प्रोडक्ट पर काम कर रहे हैं कि हमारा एनवायरनमेंट इको फ्रेंडली हो और हम ऐसे प्रोडक्ट तैयार करें। फ्लेक्सिबल मैनेजमेंट, ये लास्ट स्लाइड है ना? क्लियर है? रोहित सर पर निशानी कर रहे हैं, उस पे हमने लिखा है जो बदलता है वही दिखता है, और यही हमारा कंक्लूजन है। नीचे हमने लिखा है नए प्रबंध सिद्धांत रखने पे मनुष्य, नवाचार, डेटा, पर्यावरण और ग्राहक संतोष पर आधारित है। सफल संगठन वही है जो बदलाव का नेतृत्व करे, ना कि बदलाव का इंतजार करे। तो आपको जितना टाइम मिल रहा है, जैसा मिल रहा है, तुरंत बदलिए। नहीं बदलेंगे तो कहीं नहीं रहेंगे, और ये हमारा मैनेजमेंट भी बोला जाता है, नहीं बदलेंगे तो कहीं नहीं रहेंगे। थैंक यू।
बहुत बहुत धन्यवाद प्रोफेसर सागर सर, आपके इस ज्ञानवर्धक और मजेदार सेशन के लिए। और किसी के मन में अगर कोई सवाल, कोई क्वेरी तो पूछ सकते हैं। एनीवन? कोई सवाल नहीं? तो सर मैं आभार व्यक्त करना चाहूँगा आपका। On behalf of organizing committee, Government श्यामा प्रसाद मुखर्जी, College Sitapur, Chhattisgarh, we express profound gratitude to Professor Pradeep Jangre sir, Government Postgraduate Autonomous College, Durg. His incredibly informative, joyful session on business management in contemporary times, functions and emerging trends, masterfully connected foundational concepts like time and motions study, standardization, and modern management of Mahabharata, emphasizing harmony and training. Sir, his insights were truly enriching. Thank you, sir. नेक्स्ट जो इन्वाइटेड पर्सन है वो इंटरनेशनल साइंटिस्ट हैं, प्रोफेसर A.C. वैलेंटाइन, नाइजीरिया से हैं। Am I audible, sir? Hello?
Yeah, I can't hear you clearly, sir. Can you increase the volume, please?
A very warm welcome you to this session of the international conference organized by Govt Shyama Prasad Mukherjee College, Sitapur, Chhattisgarh, India. Sir, we are truly honored to have with us distinguished international experts join us all the way from Nigeria. Sir, please welcome Professor A.C. Valentine from the Department of Chemical Engineering, Alex Ekwemi, Federal University, Nigeria. Today he will delivering on invited talk on a very cutting edge topic, nanotechnology principle and nanomaterial in next generation corrosion control, bridging sustainability with high performance material. Please welcome, sir. It's over to you. Please welcome, sir.
Okay, good morning, sir. Good morning, all. Am I audible?
Yes, sir, yes.
Okay, please, can you upload this slide for me?
Please continue, sir.
Yeah, I'm still waiting.
Please go ahead, sir.
I've not seen the slides.
Yes, yes.
Hello?
Yes, 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 sir.
I'm still waiting, sir.
Can you see the slide, sir?
Yes, yes.
Okay, sir, go ahead.
Yes. Okay, good morning, all. Good morning. Can you hear me? Can you hear me?
Yes, sir,
Okay,
go
okay,
ahead.
Good
Go
morning.
ahead.
Good morning from Nigeria. Good afternoon from India. I'm very happy for the kind invitation from the convener and the organizing committee of this conference. I remain my humble self, Engineer Dr. Anadebechi Godde Valentine, a faculty member at the Department of Chemical Engineering, Faculty of Engineering and Technology, Alex Ekweme Federal University, Ikwalike, Nigeria. Next slide, please. The overview, we have an overview of my university. This is the pictorial view of my university where I'm working. The first one is the pictorial view of the university, and the second one is my faculty of engineering. And the third one is the top view of the entire university where I'm currently working as an assistant professor. Next slide. Here is the outline of my presentation today. My research work is focused on corrosion, just like we said earlier, the role of nanomaterial, nanotechnology principles, and nanomaterial as next generation corrosion inhibitors. This is the outline of my research. Next slide. Next slide. First, we know what corrosion is all about. Corrosion simply is defined as the chemical or electrochemical reaction, usually between a metal and its environment, that leads to deterioration of metal. In this particular presentation, we are going to focus on what we call the CO2 corrosion. CO2 corrosion is mainly seen or is domiciled in oil and gas environment. Just like we have other types of corrosion, like acid corrosion, where you talk about HCl, sulfuric acid, that are usually used for distilling and distilling. But in this regard, we are going to study the CO2 corrosion inhibition in oil and gas environment. And the materials that was used as inhibitor is called metal organic framework. So this slide is talking about the mechanism of CO2 corrosion, where a water and salt comes in contact to form a weak acid, which is called carbonic acid, that destroy the metal surface, especially pipeline steel, X65 pipeline steel or X52 pipeline steel. Next slide. These are impact of corrosion. We all know today that corrosion has contributed to serious economic damages or losses. The first slide, you can see a picture of oil and gas environment that is susceptible to corrosion. Then the second one is showing the internal pipeline corrosion of oil and gas pipeline. Then the third one is the heat exchanger. We know that copper is usually used in fabrication of heat exchangers. So this is heat exchanger, which is also susceptible to chloride attack. Then the fourth one is bridge. We know bridge that are used in sea. So those bridge or those structures are also susceptible to chloride attack while in sea, which can also result to sudden breakdown of bridges. Then the fifth one is also a copper, which are also used for heat exchanger and the rest of them. And you can see a clear image of destroyed or damaged pipeline. This is as a result of pitting corrosion. So corrosion, if corrosion is not taken care of, corrosion can lead to serious economic damage or even loss of life. So there is need to control corrosion to avoid sudden shutdown or loss of life. Next slide. You can see what we call economic burden of corrosion. This data was generated from CSI Secret India. You can see the economic data that was raised by Secret. This is an overview of the economic impact of corrosion. So according to NACE, you can see that of agriculture, industry, service. This is a comprehensive data that was taken from Secret database. Secret are the one that generated this data about the economic burden of corrosion. Next slide. When we talk about corrosion inhibitors, we all know that we have different types of corrosion inhibitors, where we talk about the anodic, we talk about the cathodic, we talk about the mixed type inhibitor. So all these type of inhibitor has its own mechanism, whether it's forming based on the metal surface, which is usually physical absorption, or whether it's chemisorption, or whether it's mixed in both. So this slide justifies or clearly gives an overview of different types of inhibitor with regards to its mechanism, whether it's cathodic, whether it's anodic, or whether it's mixed. Next slide. Like I said earlier, we have different types of inhibitor. You can see the plant extract, you can see the ionic lipid, you can see the pharmaceutical, and more recently the ZIP65, which is MOF, graphene oxide, medicine, functionalized medicine, carbon dot, CNT, and pyridine. Like I said earlier, the first type of inhibitor that was investigated as corrosion inhibitors is well known, which is called the plant extract. Plant extract has been overstudied by different scientists, but the major drawback still exists for plant extract, because plant extract contains a mix of constituents. So it's usually difficult to isolate or to identify the type or the particular or the specific constituent that is responsible for corrosion inhibitor. Quite all right, plant extracts are very good corrosion inhibitors, but the drawback still exists in identifying the active component that is responsible for that corrosion. So that is the major drawback of plant extract. Then ionic liquid is also good when you talk about the anion and the cation that comes to form the ionic liquid. Then people have also studied on pharmaceutical drugs, but it's good to use an expired drug rather than using a fresh drug. Reason for expired drug is that use of expired drug will reduce the accumulation or the generation of waste, because currently we are talking about conversion of waste to wealth. And when you use expired drug as corrosion inhibitor, it will reduce the risk of patients taking expired drug, which we know that inherently is so bad to the health. So the conversion of expired drug into wealth is a desirable development. So that's why effort has been made to convert expired drugs as corrosion inhibitor rather than using fresh drugs as inhibitor. More recently, like we said, MOF recently has gained a lot of attention due to its ability. The same thing with graphene oxide, methane, carbon dot, CNT, and other nanomaterials. So nanomaterial has gained significant attention in the areas of material science and engineering. When you talk about electroanalysis, like biosensing, catalysis, corrosion inhibitor, in fact, in electrochemistry, nanomaterial has played a vital role which aligns with the mandate and vision of green chemistry. So because of their features, the scientists were able to utilize the tunnability, the structural morphology of this material and turn them into a new materials to achieve the desirable purpose or to enhance their electroactivity. Next slide. So like I said earlier, this PPT or this presentation dwells on different metal organic framework as corrosion inhibitor for CO2 corrosion. Like I said earlier, MOFs are hybrid materials which consist of organic linkers and metal nodes. In this work, I use different precursors of metal ion and organic linker. And the organic linker I used during my work or in process of this work was 2-methyl-imidazole, because 2-methyl-imidazole is a good organic linker, is a good corrosion inhibitor. So these are the things you have to put in mind when designing a nanomaterial. What type of precursors that you are going to use. So in this case, I used cobalt, I used nickel, I used cerium, and I used zinc nitrate as a hydrate. These are the precursors I use as metal ion. Why that of organic linker, I used 2-methyl-imidazole. Why the choice of these precursors? For example, cerium MOF. Cerium has been known or is a good corrosion inhibitor for aluminium. So since it's a good corrosion inhibitor for aluminium, it was thought worthwhile that when combining cerium and 2-methyl-imidazole and the organic, that it will form a hybrid material. So, like I said, you have to consider the precursor as a good material before embarking on design of any nanomaterial. Like you know that recently MOF received a Nobel Prize because of its features, its porosity. In corrosion in design corrosion inhibitors, the porosity has a vital role to play, where you can embed it or where you can decorate other active ingredients or active corrosion inhibitor in terms of smart coating, in terms of a corrosion inhibitor release. So corrosion scientists have utilized the porous nature of metal organic framework to design those materials as corrosion inhibitor or for smart coatings. So you can see it has wide application when you talk about fuel cell membrane, when you talk about electrocatalysis, when you talk about wastewater treatment. Because of the porosity, it can be able to trap or to capture impurities and separate the impurities from the water. So you can also see that it has been used for supercapacitor and battery technology. So it has gained very wide attention in the area of material science. That's why it was able to receive that Nobel Prize recently. Next slide. Then we talk about the coordination chemistry of MOF. Just like I said, is a combination of a metal ion and an organic linker. You can use any type of organic linker, but in corrosion, we emphasize on the type of organic linker, whether the organic linker is in compatibility with the surrounding environment, the acid environment, or the metal, whether mild steel, whether aluminium, or whether copper. Sorry. So you can see the coordination, full coordination chemistry of cerium coordinating with the metal organic framework. So the metal ion coordinating with the nitrogen to form a very complex framework. It will precipitate into that framework. So this slide, this particular slide, shows the mechanism of the formation of that framework. Next slide. Then we'll talk about the synthesis of MOF and electrochemical study. Like I said, in this work, I used chemical method of synthesis. We know we have different methods of synthesis. I will say one thing here. I used chemical method, reason. I do not want to apply heat, just like normal hydrothermal. In terms of corrosion inhibition studies, it's good to synthesize your materials under room temperature, so that heat or effect, there will be no effect of heat on the material. There will be no damage on the chemical structure. For example, those in battery, those in supercapacitor or other field can synthesize more using hydrothermal, because in their study they want a smaller RCT. But in corrosion inhibition, we want a larger RCT. That large RCT will show you the corrosion resistance. So when you synthesize using hydrothermal method, there is possibility that it may affect the inhibition efficacy of the material. So that was why we decided to go for chemical method of synthesis. Chemical method of synthesis simply refers to you weigh a specific amount of your metal ion and 100 ml of your solvent, ethanol or methanol, as the case may be, and also that of the organic leather. Here I used for cobalt MOF, I used around 14 point something gram of cobalt nitrate, zinc nitrate, which was added in the 100 ml of methanol. Then also 66 gram of the organic linker. Why the 66 gram of organic linker? Is because the organic linker serves as the anchoring point on the metal surface. Why the metal ion will now enhance the framework. So that was why the organic linker was higher in terms of synthesis, to expedite the formation of the inhibitor film on the metal surface. Then we now use, after mixing, after the mixing, then the organic, the 2-methylimidazole was added in a dropwise into the metal ion solution. Then it will now give rise to this color, purple color you are seeing. And after 2 hours of stirring, continuous stirring, then we removed and kept overnight. The following day we separate. When you separate, then you go and do centrifugal process, centrifugation, to have the precipitate and your wash. Then you obtain your MOF. You can also do hydrothermal, but it depends on the application, what you want to use that material for. That's why in nanotechnology or nanomaterial, you have to consider your application first. When you know your application, you know the compatibility of the synthesis route with that material, with that application. So the next one you are seeing is the electrochemical setup. That's in our lab, that's our electrochemical setup. We are using auto lab. And the other one you are seeing there is the CO2 gas cylinder with the electrode, the complete electrode system. Then the environment I studied or I worked on is CO2 environment, like I said, 3.5 sodium chloride that was purged continuously with CO2 gas until the experiment finishes. Then the operating acid environment is 3.8, the acidic nature. And the pipeline still in question is oil and gas pipeline still, S65 pipeline still. Next slide. Then in the course of my work, I was able to develop a model, a machine learning model, to predict the inhibition efficiency. What I mean by predicting the inhibition efficiency is when you talk about any research, there's what we call the independent variables and the expected response. In corrosion, time, temperature, acid concentration, inhibitor concentration are regarded as the independent variables. Why the expected response is the inhibition efficiency. Likewise, someone that is working on battery, you are talking about your battery life, stability, time, and the rest of them. So these are the independent variables. So in my own case, inhibition efficiency is my expected response. Why those variables, time, temperature, acid concentration, inhibitor concentration, are the independent variables? Then how are those independent variables affecting the inhibition efficiency? We usually see them via experimental. So we need to understand further the mechanism using artificial intelligence machine learning model. The one I used here was called adaptive neurophysics information system modeling, that is ANFIS. So what we are seeing here is a systematic process of generating the model. You see start, you load your training. What I mean by loading the training is like when you want to build a PHK, you know your capacity, you know the amount of money you have in your account, you know the amount of plot of land you have. If that amount of plot of land can accommodate a down PHK or an upstep. So based on your data you are having, you train your data either 20-20. That 20 for validation, 20 for testing, and 60 for training, or 15 for testing, 15 for validation, 70 for training. Training must be high because training is the center point. So after that, you define your inputs. The inputs you are defining is just those independent variables I'm talking about and expected response. Then you generate the model. The model can be either multi-input. That is why I say in my own field, I have varieties of independent variables. Then I have a single output. Sorry. So my model now is what we call multi-input, single output. That is called MISO. In some cases, it can be multi-input, multi-output. In other way, it can be single input, single output, CISO. So it depends on the nature of your work or nature of your experiment. So you generate the model. Then you train the ANFIS, you test the ANFIS, and you validate. If the model did not converge, then you go back and redesign your work again. Next slide. The first work as corrosion inhibitor I will present here is serum-based MOF as corrosion inhibitor for API FLF65 pipeline still. Next slide. As you can see, the first, it's always good. Nanomaterial require an extensive, sophisticated analytical machine. You can see the XRD. You have the XRD plot of the MOF, serum MOF, SRD of the serum nitrite, hexahydrate, the precursor, and that of also the 2-methyl-imidazole. Then you can see the epicenter morphology of serum MOF. You can see the spherical ruble-like nature of that serum MOF, and also the HR10 of serum MOF. Next slide. Then we progress to electrochemical study. Just like we said, in corrosion, electrochemical study is an advanced way of evaluating corrosion inhibition. We have the traditional way, which is called the weight loss, but electrochemical is more accurate because it gives you the interfacial studies of what is happening on the metal surface. The first one you are seeing there is called the OCP. It means open circuit potential, or you can call it resting potential. Period to any electrochemical study, you conduct the OCP. OCP is just like you enter into your bike, you on your bike, you allow your bike to warm for some time before you take off. So in electrochemical study, we allow the system to stabilize at least 1 hour before you digress or you progress to polarization or electrochemical impedance spec. So from the OCP analysis, you can see that the black trend is the blank, it does not have an inhibitor. Why the other ones contain varying doses of serum MOF, so they tend towards the anodic part. Similar trend was observed in the PDP. You can see that the blank is black, showing high corrosion, whereas on addition of the inhibitor at different concentration, the corrosion current density were decreasing. So as the corrosion current density were decreasing and it was moving towards the anodic part, which also aligned with the result of OCP. If the OCP is moving towards anodic and the PDP is moving towards Catholic, that result is null and void, it's not acceptable. The information in OCP must converge, must align with that of the PDP. So OCP is moving towards the anaerobic at varying inhibited dosages. The same was seen at the PDP for inhibited cause. Then the next one you see is the impedance spectroscopy. When we talk about PDP, polarization has two things. One, there's a potential plus minus 250 millivolts and a scan rate. So polarization simply requires, as you are polarizing, you are moving, you are moving, polarizing the current, you are forcing it high. When you are moving that, there is what we call a damaged surface. Why I'm saying this is that usually reviewers will ask you, what is the distinctive difference between polarization and a Nyquist or an impedance studies? When you talk about a polarization involves that what I told you. You are polarizing, you are shifting the potential wave. Sorry. But when you talk about the impedance is a non-destructive test. It only gives you information on what is happening on the metal surface. And how do you know what is happening on the metal surface? Is by a semicircle. You can see that the blank one has a very small semicircle which is around 220 or 250. Whereas as we are adding the inhibitor, the semicircle we are increasing. The circumference of the semicircle we are increasing, showing the film formation, barrier formation on the pipeline still. So the same information the Nyquist plot is seen at the log modulus and the phase angle. Then you can see the equivalent circuit in electrochemistry. It depends, you use it to fit your data to know the accuracy of the model, the circuit model used. Also, you can see the FSM of the corroded surface. You can see different cracks, severely damaged, but on addition of inhibitor, a smooth surface emerged. Next slide. Then we talk about computational studies. Recently, computational studies have received a significant attention because it can help you to design your material, help you to know the active parts, the LUMO areas, electrophilic and the nucleophilic areas of a particular material. It also gives you insight on the theoretical background of what your experiment is all about. Just like this particular slide we are talking, remember we have done the experimental, we have done the machine learning prediction. Now we are talking about the theoretical aspect to envisage what we have seen in the lab. You can see the structure, the monolayer of how the metal, the serum was coordinating with the 2-methyl-imidazole. You can see the electron density, then you can see the HUMO and LUMO areas. Then you can see how the inhibitor, the serum was standing on the metal surface, Fe110, in CO2 environment. You can see that the serum turned very close on the surface of the metal. That is the front view. You can also see the side view. You can also see the side view, that is the figure F. You can see the top view. The top, you can see the ring, the monolayer forming a barrier on top of the metal. Next slide. Then we talk about, like we said earlier, machine learning. We employed machine learning called AMPHIS to predict what we have already obtained from the lab. Like I said, you can see the input, the left-hand side in that first diagram there is called the model design. You can see the inputs there at about 3 or 4 or something. You have the temperature, the time, the inhibitor concentration, the acid concentration, and a lot of input variables there. Then we have the rule. We have the input rule, the rule. The rule will govern what is happening within the system as you are doing your modeling, as you are doing your programming. Then we have the output, which is a single output. Like I said earlier, it's multi-input and a single output. Then the 3D graph you are seeing there shows the relationship between time and temperature, temperature and acid concentration and inhibitor concentration and time. So the yellow region are the best operating points of you can, as you can see, it have 3 distinctive layers: yellow, light blue, and dark blue. So the layer in the yellow region are the top best efficiency as predicted by this model. You can also see the predicted model because we have the experimental data and the one that was predicted by the machine learning model. That is the linear plot you have seen there. The next slide. Then that's the summary of the work. Because of time, let's move to the next slide. Then the next one is cobalt morph as corrosion inhibitor. Move on, next slide. You can see the rhombic nature from the FSM analysis. You can see the rhombic morphology of cobalt ZIP67. So that rhombic nature is a well-known defined structure when you combine zinc nitrates with any organic linker, which also you can confirm from the SHYAMA analysis. You can see the rhombic nature is clearly visible there. Then you can also see the elemental color mapping, denoting the presence or the existence of the expected element of zinc, nitrogen, carbon, oxygen, and cobalt itself. Then you can see the SRD analysis and the FTIR for the inhibited, and that of the cobalt morph, and that of the zinc and 2-methyl imidazole. Next slide. Then we go to the electrochemical analysis again. You can see the OCP tends, for that of the inhibited curves, tends towards the anodic part, while that of the blank one was a kind of a straight line. The same thing applies to that of the 2-methyl imidazole. In this case, we also tested 2-methyl imidazole alone to know if performance without being in hybrid form, what is going to do as a corrosion inhibitor. Then in the PDP, you can see that for the inhibited ones, all of them tends towards the anodic part. And that of the blank and that of the 2-methyl imidazole alone tends towards the cathodic part. Then you can see the impedance spectra, impedance studies. Like we said, the blank one has also a very small semicircle showing corrosion resistance. But on addition of inhibitor of that hybrid cobalt morph, there is an increase in charge transfer resistance. The same thing is applicable to log modules. You can see at the low frequency region, you can see there is a high value, and the same thing at the phase angle. You can see the equivalent circuit that was used to feed the experimental data. Then the next one is called the XPS analysis. XPS is a very advanced technique which can tell you what is absorbed on the metal surface. We want to confirm actually the information on the metal surface, which can also be done using FSM, quite alright. They want to go advanced by using FSM. We confirm the presence of iron, oxygen, nitrogen, carbon, and the cobalt, that they also participated as or formed a barrier film on the metal surface at different binding and energy states. That is the reason of doing SPS analysis. Next slide. Then we also did computational analysis to corroborate with the experimental findings. You can see the framework, the optimized structure of the cobalt morph ZIP67, and the electron density, and the HUMO and the LUMO diagram. In this particular one, we are able to do or to distinguish between what is happening in aqueous environment and what is happening in gaseous environment. The top view shows the side view, the top view, and the front view in aqueous environment. You can see that there is a competition between the cobalt morph and the water molecules and the corrosive environment. But in terms of the gaseous phase, the cobalt morph tends very closer to the metal surface. That means in the absence of that electrolyte, that the cobalt morph was seen tending closer to the metal surface. While in that of the aqueous environment, there is a competition, there is struggle, there is fight between the inhibitor and the corrosive environment, that is the CO2 environment. So the cobalt morph was trying to substitute the water molecules to form a barrier film on the metal surface. Next slide. Sorry. That is summary. Next slide. Then the next one we investigated was the nickel morph. We know that nickel morph is also a good precursor for catalytic process in terms of developing materials for catalytic studies. Next slide. So you can see the FSM image of nickel morph. You can see that of the nanoflakes is a kind of flake-like morphology of nickel morph. You can see the XRD, which are also, you can see the SHYAMA. Like I said, when you talk about nanotechnology, you must talk about having an extensive, a sophisticated, advanced material, which you can use to justify that this material is this. That this material has a particular structure. It has spherical structure, it has triangular structure, it has rectangular structure. So you can see that flake-like nature of nickel morph is seen in the, is confirmed by the SHYAMA analysis. You can also see the expected element. You can see the expected element of the nickel morph. Next slide. The same goes to the electrochemical. You can see the electrochemical, the OCP. The inhibited one, the same, tends towards the anodic part. The same thing with the blank one. The same thing with the blank one. The blank one tends towards the anodic part. And the same was also confirmed in the PDP polarization. All the inhibited ones at varying concentration tends toward the anodic part, showing that nickel morph is anodic inhibitor. All these morphs that have been discussed now are classified as anodic inhibitor. If it's tending towards the left-hand side, it's classified as cathodic inhibitor. If it's a mix at the center, it's inhibiting the anodic and is inhibiting the cathodic. That means it's classified as a mixed type inhibitor, having the millivolt less than 85. But clearly here, we have what we call predominant anodic effects in these morphs that we have discussed now. So we have predominant anodic effects. So the next one is the impedance study. In impedance study, you can also see that on addition of nickel morph at varying concentration, the charge transfer resistance we are increasing, showing that there is a barrier film on the metal surface which impede the permeation or the penetration of CO2 solution reaching the metal surface. Then you can see the FSM analysis. The FSM analysis shows the corroded surface without inhibitor and the ADAS. Then on addition of inhibitor, there is a smooth surface. FSM analysis go further than SEM. It gives you in-depth observation, in-depth knowledge of what is happening on the surface of your metal with good clarity. The same thing you can see for that of the atomic force microscope, that is AFM. AFM is talking about the roughness. You can see the roughness of the corroded surface and that of the inhibited surface. You can see that there is very dark pits, which corroborate with that of the corroded metal surface. Then on addition of inhibitor, there is emergence of a smooth layer showing the formation of the barrier film or formation of the nickel morph barrier film on the metal surface. Next slide. Then we also did the computational studies for the nickel morph. You can see the optimized structure, that of the humor and lumor. Then the MD simulation for the aqueous and the gas phase. So in aqueous, the nickel morph tends towards the metal surface in all the views, both the front view, the side view, and the top view. Similar trend was also observed in terms of the gaseous phase. Next slide. So next slide. Then the last but not the least is what we call the zelotic tetrazole framework, ZTF. This is a new type of morph that's just emerged. This work was published in Langmuir recently. As far as I'm concerned, or to the best of my knowledge, you can browse about this material. We have only two or three papers published in this material. It's a new type of material. This material was first designed from India with a professor in RMIT. So they use this material to work on hydrogen evolution catalytic process. That was published in I think it's ACS journal. I've forgotten what that language. I think it's in ACS journal, ZTL. So when I found this paper, I said that's good. That instead of combining zinc nitrates with that it will be good to combine zinc nitrate. It will be good to combine zinc nitrate with tetrazole as an organic linker. So I tried to combine this. In combining this, I didn't use chemical method. I used mechanochemical method because in their study they use mechanochemical method. When it comes to nanomaterial synthesis, your synthesis route matters a lot. You can use chemical method when you get another morphology. You can use mechanochemical, you get another morphology. So it's very important to know your application and know your material. Then you know the best synthesis route that best suits the process. In this work, I used mechanochemical method of process. So in the absence of solvent, other ones, you remember I was using methanol or ethanol, 100 ml, chemical method. But in this process, I will bring zinc acetate, I will bring tetrazole framework, organic linker, and grind them mechanically. Next slide, you see the synthesis route. Yeah, see the synthesis method. Zinc acetate combined with 5-methyl tetrazole as organic. Then we added sodium hydroxide as a stabilizer. Then we mixed. When we mix, we put in the this thing and put in the oven for 12 hours. For 24 hours under 70 degrees. Then the next day you bring, you wash and you dry. The temperature we use is 70 degrees. It's not high, so it does not have any effect on the material. Next slide. So this is called mechanochemical method. Next slide. Then you can see the morphology, the FSM morphology of ZTF8. It has almost 6 sides. It has almost 6 sides. We call it a tetrahedral shape. Then you can also see it clearly from the HL10. HL10 also gives the same image we obtained from FSM. You can see the SRD. The SRD almost have the same with that of ZIP8. So they have the same pattern. Then you can see that of the FTIR analysis of the material. Next slide. You can see the electrochemical studies. In impedance studies, it's the same. You have to combine the control is of blank, and the inhibited ones the other semicircle will be your inhibited ones at varying concentration of your inhibitor. So you can see the control, which is the blank, it should be around 220, showing the corrosion, showing corrosion. But as the inhibitor, as ZTF was added at varying concentration, you can see there is an increase in charge transfer resistance. Similar thing is observed at the log modulus plug and that of the phase angle. You can see the equivalent circuit, a 2-time constant equivalent circuit that was used. In all these studies, you can notice that 2 times equivalent circuit were used. Why? The reason for using the 2-time constant equivalent circuit is because of the porosity nature of mode. There is tendency to form a second film. There is a tendency to form a second. Any porous material you are using as corrosion inhibitor, you must fit it with a 2-time constant because you are giving kudos to the first CPE 1 and the second CPE 2 due to the formation of the first barrier film and that of the second one because of the porosity nature of the material. Then in this one we also did the calorimetry test, a kind of optical test to also visualize the roughness of the corroded and that of the inhibitor. You can see the clear image of confirmed CO2 corrosion. of X65 pipeline still. Then on addition of the inhibitor, you can see that the smooth and a clear image of the inhibited one emerged, showing the barrier formation of ZTF on the metal surface. Next slide. Then you can see the XPS analysis. We use XPS to confirm the absorption of this barrier film on the metal surface. You can see the sulfur, the carbon, the nitrogen, the oxygen, the iron, and the zinc element. These are elements we are confirmed on the metal surface that they absorb on the metal surface at different oxidation and binding energy. Next slide. Then we also did the computational. You can see the optimized structure of the ZTF8, the electron density, the HOMO, LUMO. Then, and the MD simulation. MD simulation talks about the interaction. You want to know the absorption orientation on the inhibitor on the metal surface. We also have that Monte Carlo simulation. But MC simulation will tell you the surface coverage, the pattern of surface coverage, how the surface, the amount of area that was covered by the inhibitor. That's the clear difference between MC and MD. MD will tell you the absorption orientation, absorption pattern on the metal surface. That of the MC will tell you the coverage. It has to do with coverage. What area was covered by the inhibitor? Sorry. So in this one, you see in the absence of in gaseous phase, the ZTF is very close, very close on the metal surface. While in that of the aqueous environment, there is severe competition, there is struggle. The ZTF was struggling to substitute, to force himself on the metal surface. So there is a little gap between the metal and the inhibitor. But in that of the gaseous phase, they tend very close. They tend very close. So that is why it's always good to perform your modeling in gaseous phase without a corrosive environment and also with corrosive environment. Next slide. And that's the summary of the work. Next slide. That's the summary of the work. Summary of the work we have just discussed now. We can see that cobalt morph has better performance. Even when we did the stability study was more than 24 hours. So it has higher performance than other morph that I have studied so far. Then next slide. Then these are a few publications that emerged from the work so far. Next slide. Yeah, I want to acknowledge my former director that welcomed me in India, Sikkim. First and foremost, I always tell my friend Dr. D.K. Verma that India is my second home because I was in India for complete 4 years. In Tamil Nadu, I was able to learn some cultures, some food, and other things. I have friends. So I want to acknowledge Dr. Kalai Slavi, which is currently also the DG CSIR. She was the one that welcomed me in Sikkim. And also the current director, Dr. K. Ramesha, is doing a good work, he's doing a great job in Sikkim. And I want to acknowledge CSIR, Council for Scientific and Industrial Research India, because they are the one that trained me. So I'm also TIWAS, the World Academy of Science. The World Academy of Science, TIWAS. TIWAS and the government of India have a good rapport or a good understanding to form this fellowship for developing countries, which I benefited from. So I acknowledge the government of India and that of TIWAS, Italy, because all this work would not have been possible without coming to Sikkim, CSI Sikkim, India. And also I acknowledge the support of my boss, Dr. Rakesh Barry, who also is the current head of Department Corrosion and Material Protection Division of Sikkim. I also acknowledge the support a friend, a brother, Dr. D.K. Farmer. He is a pity that I have not seen him facially during my stay in India. Then we are always in contact, and he's the one that gave me this contact to attend this conference. I appreciate him, brotherly, I appreciate you. I also appreciate the convener and the organizing committee of this conference for finding me worthy to come and present my work in this conference. Thank you so much. I remain blessed. Thank you.
Am I audible, sir?
If you have questions, you can. Next slide. If you have questions, you can ask.
Valentine, sir, am I audible? Hello?
So I'm not hearing you clearly, please. Can you increase your volume?
Can you hear me, sir?
Yes, I can hear you a bit now.
Okay, thank you so much, sir. Your wonderful session. Is there any question from audience? Any query, please? Feel free to ask. पूछ सकते हैं आप? Valentine, sir, is there
yes, sir.
No questions? Is there no questions?
Okay,
No
okay,
worry.
sir.
Then I vote of thanks on behalf of organizing committee of government S.P.M. College Sitapur, Surguja, Chhattisgarh, India. I extend our profound gratitude to our esteemed international speaker, Professor A.C. Valentine from the Department of Chemical Engineering, Alex Ekwimi, Federal University, Nigeria. Professor Valentine, your illuminating lecture on nanotechnology principle and nanomaterial in next generation corrosion control, bridging sustainability with high performance material was truly exceptional. You masterfully highlighted how nanotechnology is revolutionizing corrosion control, offering sustainable solution with high performance material, a topic incredibly permanent to our international conference on emerging trend in science, technology and management. Thank you once again, Professor.
Thank you, sir.
Chiga, sir.
Thank you, sir. Thank you, sir. You're welcome. Thank you, sir. Thank you, sir. Thank you, sir. All right. Bye. Bye. Bye.
Bye, sir.
Bye, sir. Yeah.
Thank you, sir. With this, we conclude our technical session. Now there is a lunch break of 45 minutes. It is 2:15 to 60, and we will join for the next technical session by 3 p.m. Thank you.